because plant has no choice. So wherever a seed fall or planted, it is going to respond to that soil, water availability, availability of even the wind, and obviously the light. So then and there the decision is made how the seed is going to germinate and how it's going to grow, whether it grows into a is there a pointer in there? Uh, that's in the, okay. So whether it germinate into a full growth or barely make it or somewhere in between is a function of where that seed happened to fall. So it's simply responding to that environment. Now, because the environment affects all the way from germination to the flowering and seeding, ultimately harvesting the crop. So this continuous effect of environment really has to be titrated, and we harvest that in the end, what will be the yield or so-called harvest index, what you are producing out of that given plant. So since plant has to make those decisions for survival and full productivity, if those decisions are not made right, then the plant is going to die. So this is a do or die situation for the plant to handle the environment. So one of the things that happens, environment slows growth. Now we all know in adverse environment, if you don't water the crop, it's not going to grow as well. Plants have to then adapt to that slow growth, and if that environment persists for a long time, then the mutation might occur, and then it may convert into a race. So particular race of the plant are more suitable to grow in a particular environment, while others cannot from the same species. And eventually it leads to speciation. So these are gradual process, time dependent process, and plant has to continuously make those decisions. Now this condition, that is the stress, is actually inherited. So there is a memory in the plant which knows that I have been through such and such a stress, and that memory is inherited to the next generation in order to perform better during the next generation. And it's also true for humans. It is not only related to the plant. So original information was actually gleaned from the famine and feast that have gone through hundreds of years back. An analysis of data gave some clue that something is going on in terms of inheriting certain traits that allow a particular group to survive under adverse conditions. So this generationality is really can last for 2 to 3 generations, even under the optimum growth conditions. So it is erasable memory. It is not a permanent memory. So you can erase it, but it takes time to erase that memory. That's not quickly erased. So this brings you this 18th century French scientist Lamarck, who had predicted all this way before anything we knew about gene or genetics. It's way before that. But that idea was rejected because there was no real basis for how to explain that. But Lamarck was very wise kind of a person who intuitively came up with this idea, and it was later followed by Lysenko in Russia, which said that if you grow plants under certain nutritional conditions of high nitrogen or phosphate, they carry certain memory in the next generation. And that idea was boo-booed because, again, there was no real genetics at that time, and Russian society was very controlled at that time, so it was really not followed up at all. And they suffered as a result of that because they didn't allow certain aspect of genetics to really flourish. So, what is a stress? Stress comes in a variety of forms. Drought and salinity, we know commonly the plants need water, or you keep on irrigating and evaporating water, the more salinity builds up in the soil. So these are so-called abiotic factors really affecting osmotic stress. So the osmotic stress is sensed by the plant cell, and that affects the growth. So growth is reduced, and if growth is reduced, the productivity will be reduced because these are directly proportional. So there is a example of this very well done in a Japanese lab where they were able to harvest 3,000 kilogram of tomato from a single plant. So how they did it is providing the right kind of nutrition, environment, and they fully exploited the complete genetic potential of the plant. One plant, one season. Yeah. So that shows you that one can indeed grow things to its full potential provided you give what the plant needs. So, stress leading to growth alteration, we know, even the farmer knows that. But there's a big black box in between. From a scientist's point of view, what is that mechanism that allow a stress signal to interface with the growth signal? So these are basic cellular machineries. They have to communicate with each other and then perform to the best of the potential that exists. So growth requires protein synthesis. Protein synthesis then lead to the cell division, and it controls obviously the gene expression. However, what I'm going to tell you this morning is that protein synthesis itself controls gene expression. So there's a feedback loop which tells these machineries, these are independent machineries, as like an assembly line. So the gene expression is one machinery in itself. It just makes the RNA. That RNA is then translated, make protein, protein does its function. But the rate of synthesis dictates in a feedback manner how the rate of transcription is going to go. Because if it goes too fast or too slow, you will pile up the intermediates. So these two things have to be tightly linked. So you have basically, sorry. So osmotic sensing to growth has to go through protein synthesis, but in between you have a huge effect of osmotic stress on the energy supply. And that shock, so-called of stress, has to be mediated. This is like putting a bandage on a bleeding wound. So plant produces a variety of osmoprotectants, and within those osmoprotectants, proline is one amino acid, which pathway of that particular amino acid was discovered in my lab. So we figured out how the proline is made from glutamate, and then discovered a new enzyme, P5C synthase, which take glutamate into P5C, and P5C is reduced into proline. So that enzyme P5CS was a quite unique enzyme. It was feedback inhibited by the end product, the classical bacterial scenario. So we figured out how the feedback inhibition of that enzyme is working, and we site direct mutagenized that particular inhibition site. So the enzyme was no longer inhibited by proline. And that is where Professor Kavi Kishore joined my lab for a couple of years. So he took that gene and put it into plants to demonstrate that those plants carrying that mutated P5CS can indeed make much more proline than the normal plant or the normal enzyme without mutation. So that was very important piece of work, and it was very highly cited. Even today, that paper is very much cited for this work. So these osmoprotect, proline is not the only one. There are many osmoprotectants out there, and they give you temporary relief, so-called, from the stress. So if you have these osmoprotectants, then the growth will occur, although not as good as the without stress, but not as bad as without osmoprotectant. So they are sort of ameliorating the severity of the stress. Can you move the slide, please? So, beside the abiotic stress, you have biotic stress, various insects, fungi, bacteria, they all attack the plant, so they create biotic stress. Then you have nutritional stress, and there are a variety of other signals. They create a reactive oxygen species, particularly photosynthesis and respiration being affected. So these variety of stresses continuously sending signal. But how does the plant know which signal to respond first? Because if you have 5 things happening at the same time, something has to be prioritized. You look at a parabolic antenna, TV antenna in your house, you have satellite signals bouncing on it, but the channel is tuned to one signal, so it's going to only process that signal further down. So that prioritization of the signal is the key, because the decision making has to take place somewhere. So how that works? So when you have a situation where you have to make the decision or you're going to die, then plant really deals with first thing first, and that is energy consuming processes. The protein synthesis consumes the most energy in the cell. Almost 50% of cellular energy goes in protein synthesis. So you have to slow down protein synthesis machinery immediately in response to the stress. So you can't wait for any mutations, because those are very slow long-term processes. So there has to be alternate mechanism to deal with the stress then and there and adjust the rates of the protein synthesis machinery and the transcription machineries. So these are energy consuming processes, and downstream then they produce specific enzymes which are needed for growth under a given condition. So these stress signal, they have to be prioritized as I mentioned, and then they regulate transcription and translation processes. So you have to somehow link the stress signal coming from one end and the growth which requires cell division is on the other hand. So you have a basically significant amount of communication going on between the these different processes continuously. So linking these two types of signals, first we have to understand what these signals are and how they are prioritized and how they are really communicating to the translation and transcription machineries. Only then you can understand how the growth will occur under certain conditions. So this complexity is very high because you have variety of stress signals in the first place, and then they all generate something called MAP kinase cascade. So this is a cascade which really takes the signal down through this MAP kinase pathways, and all these signals then converge into this TOR kinase, which I'm going to talk about in more detail. So the TOR kinase is really a kind of a brain of the cell. It makes the decision which way to go, how to prioritize, and how to send the signal downstream. Next slide, please. So, what is TOR? TOR is a target of rapamycin. The rapamycin is a drug that was discovered in a Pacific island, Rapa Nui, and secreted by a fungus. So this drug was used as an immunosuppressant for organ transplant to suppress the immunity. That was the primary use of that drug. But the actual mechanism was not known. So as always using model system as yeast, initial work was done in yeast, and then a homologue of this was found in animals. And being interested in stress biology, I thought there should be a plant homologue for TOR as well. So I embarked upon hunting for this molecule in plants. Next slide, please. So the TOR is really a kind of a master regulator because it controls many different pathways downstream. It responds to stress, obviously. It is also dealing with ATP homeostasis, so it's titrating the energy level in the cell, and nutrition, hormones, and raw signals. So all these parameters are being titrated all the time. Hence, the cell can make a decision based on the signal coming from the TOR, what to do and what not to do at a given time. So TOR has its footprint into translation, transcription, ribosome biogenesis, cytoskeleton, protein kinase C, even taking the cell out of the stationary phase, autophagy, and other transport systems in the cell. So it's a very complex pathway, but it is needed because you are dealing with so many parameters at the same time. So we discovered that plant contain a complete TOR kinase pathway, very similar to yeast or human, and this pathway is regulated by a single gene. So unlike yeast where you have two genes for TOR, plants have only single gene. So if you mutate this gene, the lethal is a lethal situation. So plant cannot grow without TOR activity, hence plant is essential for growth. So this kinase uh, actually bind to rapamycin. So rapamycin is a macrolide molecule that uh, binds to this domain of the TOR, sandwiched by FKBP12, another 12 KD protein, which actually binds the rapamycin, and then you bind the FRB domain of TOR. So this is a large enzyme having variety of domains. It means providing enough space out there for interaction of all kinds of adapter molecules. So it has a very large sort of a footprint, and the enzyme itself is sitting inside another basket which we call is raptor. So TOR is sitting in the raptor's basket, and then these two enzymes communicate and send the signal down. So interfacing this energy and nitrogen metabolism to growth, it allows the plant to survive under stress. So understanding this pathway is actually very important, how we should move forward to manipulate genes for stress tolerance. So if you slow down protein synthesis, you are actually slowing down cell division. Accordingly, you are reducing the body size, and plant has another sensory system, that is, if there is a stress, that it wants to quickly complete the life cycle. It doesn't want to wait and die, which makes sense. So in the process of doing so, it doesn't have much time to grow vegetatively much and immediately start flowering, and whether it can make one flower or one seed, but one seed is better than no seed. So it tries to complete the life cycle as soon as possible. So as I said, the footprint of this enzyme is very large, and it was studied in yeast using chemical genomics. So chemical genomics really allows you to mimic the signal. So treating the cells with rapamycin, you can look at the transcriptome down there and look at which proteins are being affected as a result of rapamycin treatment. So we identified almost 1200 genes which are analogous or homologous to the yeast situation that are affected by rapamycin. So experimentally we also demonstrated that TOR is interacting with raptor, and then raptor is actually communicating with S6 kinase. So S6 kinase was known way before we knew anything about TOR. We knew under stress that one of the ribosomal protein get phosphorylated, and that phosphorylation was done by S6 kinase. This much we knew, but we did not know the linkage between these different players. So using a kind of interaction assay, we were able to show the raptor interact with S6 kinase, and the immunoprecipitated or co-precipitated material has actually the kinase activity. So this was experimentally confirmed. Then S6 kinase further has its target, and the target is ribosomal protein S6. So on a small ribosomal subunit, there is a S6 protein which gets phosphorylated. So you have now a pathway of signal from outer environment going directly onto the translation machinery, and that signal basically controls the rate of translation. Once the ribosomal protein get phosphorylated, now it can slow down or accelerate the the translation machinery. So the rate of translation is immediately controlled in response to that signal. So the question was, what happens then? And one of the graduate students in my lab did the basically immunoprecipitation assay using ribosomal protein antibodies, and then looking at what else come down with it. And number of proteins came down, but among that, this HD2B, which is a transcriptional activator, or repressor rather, that caught my attention. I say, oh, there's something going on here. The protein is phosphorylated and it's now talking to a transcriptional regulator. So we followed that up and we demonstrated that histone deacetylase 2B actually interact with ribosomal protein S6, and this complex then moves from the cytoplasm to the nucleus, and in the nucleus it goes to nucleolus. Why nucleolus? Because nucleolus is the site where all the ribosomal genes are. And when you're slowing down protein synthesis, you don't want more ribosomal RNA to come, because you don't need ribosomes. You're already slowing down the protein synthesis machinery. So this is a kind of a feedback mechanism to control the rate of transcription of ribosomal DNA, and we physically demonstrated that this complex, histone deacetylase 2 and RPS6, actually binds to the promoter region of the ribosomal DNA. So we physically carried out those experiments, and we localize or co-localize histone deacetylase 2 and RPS6 inside the nucleolus using this BIPC assay. So this is experimental evidence that indeed the two molecules are together and being co-localized inside the nucleolus. And HD2B was already known to be in the nucleolus from a previous work, so this makes all the sense, and we actually mapped onto the gene where this binding occurs on the promoter of the ribosomal RNA gene. So HD2B is required to regulate transcription of ribosomal DNA after interacting with ribosomal protein S6, and this is actually a mechanism by which you can open or close the chromatin where the transcription can begin in an open chromatin, or the transcription closes and the chromatin condenses. So the histones play a very important role, and acetylation and deacetylation of histone is the key for the chromatin condensation. So these things go hand in hand, and thereby one can look at how or what rate of expression particular gene is going through. If the rate is high, you are going to make clearly more protein. If the rate is low, you are going to make less protein. But there is much more than this. It's not a one-to-one relationship. The rate of transcription actually can dictate whether you are going to do alternate splicing, hence the change the character of the product completely. So, rate of transcription affecting the end product. What are you going to make from the same gene? So, gene is still there, but the splicing of the RNA can alter depending on the rate of transcription. Hence, the product can be very different. So, rate of transcription may control type of transcript, and alternate splicing can change the entire proteome. So you have a very significant consequence of this rate. So these two processes clearly are tightly linked, and they are continuous, keeping in mind that protein synthesis consume almost 50% of cellular energy, as I mentioned, and under stress, energy is immediately affected. So it is this situation so you have basically from the upstream signals feeding into the TOR kinase, you have downstream effectors that are being affected as a result of all this process, because you are technically affecting entire transcriptome and proteome of the cell as a result of the rates of transcription and translation. So now you have a physical link where you can study and manipulate how this environmental signal going through the MAP kinase cascade, feeding through the STOR kinase, going to the S6 kinase, phosphorylating ribosomal protein, which in turn then binding to the histone deacetylase and controlling the transcription. So this is the circuit basically that we worked out, and this is quite intriguing because it makes perfect sense that environment has to be sensed and dealt with then and there. You cannot wait and see, because the damage could be irreversible to the cell and cell may die in the meantime. So you have now a link between the stress signal, the MAP kinase, the TOR kinase, the translation, transcription, and then not only ribosomal RNA, but other genes are similarly controlled by different kind of histone deacetylases. So this is only one example I gave you, but there are other histone deacetylases which have been shown to control a transcription of other genes. So this is basically talking back to the chromatin, thereby if the signal persists for too long, it can create an epigenetic mark onto that chromatin, because it's not a transient situation, it's much longer, and you don't want the cell to quickly revert back. You want that imprint onto the DNA. So that is where the epigenetic mark is left on a given gene, and this is clearly shown that if we make antisense of histone deacetylase, it creates developmental abnormality in the cell. So leaving the epigenetic mark requires methylation of DNA, because it's the methylation signal that is put onto the DNA, and histone deacetylase 2 actually is a part of the repression complex. The replication complex is much larger than just HDA2. It is literally several dozen proteins involved in this repression scenario. Histone deacetylase is only one of them. So this modification really leaves the epigenetic signature by methylating lysine 9 residue onto the histone, and then that bind to the complex, then it recruit methyltransferases. As yesterday you heard what methyltransferases do to put signature onto the DNA. So this is a clear link which really either open the chromatin for transcription by removing, by adding acetyl group, or by removing acetyl group you make a condensed chromatin. So opening and closing of the chromatin is required for transcription to occur. Condensed chromatin cannot be transcribed. You have to open the chromatin and then you carry out the transcription. So this is a basically switch between HDA, histone deacetylase, and histone acetylase. So you can actually measure these two enzymes being directly related to growth or resting state of the cell, and that has been done. So you can see that actively growing cells have more acetylation of histone, hence more open chromatin, but resting cells have condensed chromatin, and hence you have repression there. So acetylation and denaturalization are basically two functions that allows the cell to respond to the external signal and thereby controlling rate of transcription as the signal persists. So that is not the only story, because this signal can work at the post-transcription level, and post-transcription level is basically changing in the RNA stability. So that is a post-transcriptional state. So not only the rate of transcription is affected, you are now affecting the rate of RNA stability. So variety of developmental programs, including vernalization in plants, are actually epigenetically controlled. So epigenetic footprint is very large on the genome, and since it can go back and forth, it allows basically interface with the environment, and thereby plant can respond to good or bad environment immediately. Now, besides just simple growth, the epiphany of epigenetics goes beyond, and it is really true that plant will be including the higher animal. If it is true for the plant, it would be true for the higher animals as well. So you must have heard this saying in Vedic literature, you are what you eat, and that is now becoming almost true, that more and more populations getting diabetic because the pattern of the food has changed, hence its imprint onto the population has changed. So if you look 100-year-old, even 50-year-old data on the human population, completely different rate of diabetes as compared to today, because the food chain is completely altered. So if it is true for these kind of obvious traits, then one can stipulate that other characters could be epigenetically controlled. But that becomes a speculative situation, and one doesn't want to go there because we have no data, but one can clearly surmise that there could be other traits that are controlled through epigenetics. So I will leave it here and take some questions.
Thank you very much.
Yeah.
So it's a very wonderful talk.
Yeah.
And so now the talk is open for discussion. You have any questions, please?
Give him the mic.
In fact, it is just a comment and compliments to Professor Verma. I have listened to many eminent speakers about how human behavior is there under stress conditions, but the way he has explained how plants respond to stress and how the protein synthesis or translation or transcription factors are influenced, leading to the possibility of inheritance of the stress related modifications in the gene compositions in the next generation is wonderful.
Thank you.
And I would say that this has added a lot of knowledge to us, your presentation. Compliments to you.
Thank you. Thank you. Yeah.
I have two questions.
Yeah, go ahead.
Why is this memory, stress memory, only for 2 or 3 generations, and what controls that?
Well, that is basically to maintain the imprint, you have to have a continuous feedback. So if stress is removed and the conditions are normal, then that methylation comes off basically.
And obviously there must be demethylation.
Sure, demethyl. That demethylating enzymes, yeah.
Okay,
Yeah.
all right, thank you.
Thank you very much, Professor Verma. Always a learning experience from you, sir.
Thank you.
So I've always wondered, taking this into cognizance, plant exomes have just begun, in taking up, to the next level.
Yeah.
How far are we in understanding the kind of, abiotic stress spectrum, so that the plant exomes can probably herald the future genomics?
There is no information in that direction.
Yeah.
That's open field completely for you guys to study.
That was.
Yeah.
Public question.
Yeah, go ahead. Yeah.
They are talking about speed breeding these days.
Street breeding. Speed breeding, yes. Yeah.
One crop you can 3 times you grow. What happens if genetics?
Well, only time will tell. The entire agriculture system is man-made, as you know. We didn't have wheat, we didn't have corn, we didn't have any fruits. So entire genetics of the food is all man-made in last 100 years or so. So now if you speed breed, you're going to eliminate certain traits because those traits require a space. Let me give you an example for that. That's a very important question. Because there are genes that delay the onset of the next stage. For example, there's a gene called EMF, terminal flower. So if you mutate the gene, the seedling can immediately flower and seed. But you have no leaf, no energy supply, hence how can you support the seed? It's like a baby having a baby. So this kind of a scenario is programmed in the development that don't bring the next step unless this is completed. And this is universal in all cellular processes. Just take a cell division, simple step. You have karyokinesis, then you have cytokinesis. So the cytokinesis will not occur unless the karyokinesis is completed, and even within the cytokinesis, the cell plate will not complete unless the previous step is. So there's a check and balance continuously in the cell to ensure that previous step is completed. It's an amazing kind of a precision in the cellular machinery, because you got to walk, you cannot jump there. Speed breeding is just the word right now. I mean, you are basically developing crops that can early flower and seed. Yeah, so those things are going on. There are many crops that have been breeded to adjust to the different climatic conditions. So this will continue, particularly for vegetable crops, which can be grown under greenhouse environment. So that's a control environment altogether. But in nature, it has to take the same cycle. Hence, you cannot do that for outside cropping. Yeah, yeah.
My question is, in the last slide,
Yeah.
you tend to imply, so you tend to imply that somehow green revolution is related to lifestyle diseases.
Correct. Yeah, because green revolution means plenty of food. The plenty of food means plenty of consumption. So consumption is our decision. Just take example of sugar. Sugar is a man-made thing. 200 years ago, there was no rasgulla and mithai to go and get it. There was no sugar there, basically. So all these things are, with time, have been developed, and being what humans are, they go to the easiest path. The sweet is sweet, so you just consume it without thinking what's going to happen tomorrow. And consequences always come. So yeah.
So, good morning, sir.
Yeah.
Sir,
You talked a lot of kinases, and what about phosphatases? Because the balancing between the kinases and phosphatases is the whole signal transduction pathway. I didn't hear any phosphatases role here. Phosphatase.
Oh, phosphatase. No, phosphatase does have a role. I did not talk about it because all that signaling that's coming down is being titrated. So if you have too much signal, even in the TOR kinase, because it's a big molecule, so it's loaded with various kinase sites, and those kinase sites are titrated, particularly with PP2, and that is determining how to move forward, because this signaling process is a balancing act. After 5-phosphate or 10-phosphate being added, this will happen. So it's not a linear process, it's a cascade. Yeah. Yes, also the insulin phosphate, yes.
good morning, sir.
Yeah.
Sir, my question is there any link between S6 kinases and ubiquitin system? Because we have to maintain the balance between synthesis and degradation. When we are totally stopping the synthesis of protein synthesis we are stopping, then even we have to control the degradation process also.
Correct.
So is there any link between the ubiquitin and S6 or proteasome and S6 kinase?
Correct. That signal is a separate signal where you want to get rid of something that's no longer needed, otherwise going to linger. So ubiquitin-mediated pathway is a cleaning pathway. Things have happened, let's clean the house. Otherwise, the dirt will linger there. So this is a different mechanism altogether. Yeah.
Yeah, 3 of my teachers are going to give their talks today. One of them is Professor Deshpal Verma, with whom I happen to work, at Ohio State University, Columbus for 2.5 years or so. And even at the age of 80, look at the energy that he has. Even today, he goes to the department by 7 o'clock and he works there till 8 o'clock, 9 o'clock, 10 o'clock. always, there are 20 postdocs or graduate students working in his laboratory and working day, night, day out, And, he was the first person to clone several of the genes and discover the pathways. And one of them is proline biosynthetic pathways. And we get a lot of citations, I happen to work on proline biosynthetic pathway, and we get a lot of citations for that one, And thank you very much, sir, for giving me an opportunity. And thank you very much for coming all the way from Ohio to this place. And then the second talk is about Dr. Nese Srinivasulu from International Rice Research Institute, Philippines. He is also my teacher. I happen to work with him. And the next talk is by Dr. Srinivasulu. And third, my teacher is also here in front of me. That is Professor Ratnagiri. With all of them, I happen to work for some time as postdoc. Look at those people. We need to imbibe those qualities. 10 o'clock is too early for you to get up. But they get up at 2 o'clock. And he sends me a message at 2:30 or 3 o'clock. And these people, they send messages 3 o'clock or 4 o'clock. But you don't get up even at 10 o'clock. What is this? You need to imbibe such things, such qualities, all the students especially. Thank you so much.
Anyway, thank you, thank you.
Thank you very much.
Okay.
And sir, I just before closing this talk, I just have a very general question to you. I also to have, and it's a very inspiring talk. I'm
Thank
very
you.
much impressed. And very long time back, I worked with Professor Nario Murata.
Oh, okay.
So it's on cyanobacteria.
Yeah, I know that, yeah,
know him
yeah.
probably. So it's a very simple signaling system. There
Yes.
histidine kinase and
Yes.
response regulator
Yeah.
regulating the gene expression
Correct.
under different abiotic stresses. Even when it comes to the control of translation, this is which is
Much
very
simpler, yeah,
important,
yeah.
very important because they don't want to spend a lot of ATP energy
Correct.
for translation. So simple ribosomal modulating factors are
Can make
enough.
a big difference.
Yeah, just they bind and then control the translation.
Yeah.
And here in this case, a lot of
complexity,
MAP kinase
yeah,
cascade
yeah,
system
because-
and then TAR you are introducing today. To me at least, I mean, I was not aware of this TAR. And now, why this complex system has been evolved? Why do we need
That's a good question. That's a very good question. As the organisms have evolved, the complexity evolved in parallel, because the multicellular organisms have to deal with variety of issues, all the way from the amount of growth, the amount of seed production, the dealing with all the stresses. So when you have all these factors to deal with, you got to be prepared. With one weapon cannot deal with all the issues. So you have multiple weapons ready to fight. It's a war going on there, literally.
So is there some microRNAs also involved?
Correct,
in
absolutely, absolutely, because the continuous biotic stress and that signaling pathway is completely different, where the organism is continuously dealing with the pathogens, cutting and attacking, and so that's another, I did not bring that complexity in, but that's there.
Very simple last one. I'm sorry, I'm taking more time because I'm excited about it. So now the start. Can we use this as a target, genetically modified to improve? Because agriculturally,
Correct.
can we use this
Yes.
for improving
So
the
that's
stress
a
tolerance?
good question. Can one use the TOR as a tool? The experiments have been done to overexpress TOR activity, and that is directly proportional to growth.
Okay.
So it means there is a reciprocity between the growth and the level of the TOR in the cell. So one can take it as a starting point and play around with. The one thing I did not mention, the TOR being what it is, a very large molecule, is actually one of the largest gene in the cell. It's about 17.5 kb. It took us 5 years to put it together just to isolate this gene. You can do it easily now, but in those days we had to put pieces together to make this whole transcript functional. But it has so-called adapter sites. So there's a protein called LST8, which we also did some work I didn't talk about. So LST8 is like a seesaw. So under favorable condition, you got to modulate the gene. And LST8 itself responding to hormones. So you have now a complete link between the growth and the stress. Seesaw.
Thank you very much, sir. Thank you very much. I'm really inspired
Yeah,
by your talk. And a big applause. And on behalf of the organizers, I really thank you very much for a nice talk. Our next speaker. Dr. Nisse Srinivasalu, our next speaker, is going to talk about consumer-driven grain quality and nutrition. International Rice Research Institute is from. So may I invite him to deliver his talk, please? It's online. Okay. Okay, it's online talk.
Great. Oh, thank you so much. So can I just check briefly whether you could see my screen as well as can you hear me?
No. Wait. Check what is the problem. Just wait, please. It's not coming here.
How about now? Can you see?
No, not it. I mean, they're trying. They are attempting. Our technical team. Just a few minutes, please. Connected? Okay, now you have to that one. Yeah. Share now. What's the problem? ఎవరన్నా ఉన్నారా టెక్నికల్ ఎవరన్నా ఉన్నారా Don't worry, you disconnect this so that it will connect. Okay. You disconnect this one from this and directly connect to it. Maybe because of multiple. It's now coming. He has to share the screen. Is he sharing the screen?
మీటింగ్ ఎండ్ చేశారు మీటింగ్ వస్తుంది సార్
కాదు వినండి
మీరు ఎండ్ కొట్టారు
షేర్ చేయాలి స్క్రీన్ మీరు షేర్ చేయకూడదు కాదు It's okay. Now, yeah, it's now connected. We are able to see your what is it, presentation. You may please proceed now.
How about my audio?
Yeah, yeah, you are audible.
Wonderful, wonderful.
Please.
So first of all, I really like to kind of thank Professor Kavikishore for this great opportunity to kind of highlight some of our recent work. And again, I was very, very delighted to see Professor Verma's talk. At the end of his talk, he has given a very clear pitch how important it is in terms of food what we consume is really going to shape our lives. So I think building on that, what I would really like to kind of highlight today is as a staple rice, which is kind of currently being used as an important crop in India, elsewhere in Asia, how that is going to be critical to ensure energy requirements and sustain food security, but also what are we doing in terms of nutritional requirement. So can some of these cereal crops, including rice, provide both food and nutritional security in future? Or what are the alternative options we need to think through to kind of really address the double burden nutrition problem? So my name is Nesesh Srinivaslu. I'm leading grain quality and nutrition program at International Rice Research Institute. And basically the mission of IRI is about assuring global rice supplies as well as overcoming the poverty and hunger and improve health of rice farmers and consumers with environmental sustainability. So those are clearly our institutional mission and mandate. And quite lately, we have actually established a regional center in India with a very focus that we really wanted to bring in the technology of breeding and then create a value around through IRI South Asia Regional Center, where the center has been established with the clear mandate that increasing farmers' income by stabilizing the rice productivity as well as addressing the value addition and any diversification opportunities what we might have. So from that perspective, basically the host of technologies what we are bringing in India at ISARC has given a various possibility to link all the way breeding nutritious rice varieties to making value-added food products. And so that's actually a kind of latest development from our institution. So at present, currently, we are facing really the double burden nutrition problem. On one hand, nearly 2 million people, they're suffering from lack of either iron, zinc, vitamin A, folic acid, or iodine. On the other hand, equal number of people, have been basically suffering with increased obesity and non-communicable diseases. And this was really not the case in 1950s and 60s. But right now, what we see is both dimensions of double burden nutrition problem is coexisting. And the threshold could be very different in the rural areas versus urban areas. And this is where I think it's really going to be important for us to think through bringing in the quality of food in our plates and making sure that not just calories, but also nutrient-dense food we consume. So quite often when we really look from that perspective, it's really important to, it's really quite important actually to see that the rising obesity as well as diabetes has been really the major problem. And as of now, nearly 592 million of diabetic people were projected by 2035. And as it stands right now, we have seen that more than 440 million people, they are type 2 diabetic, and predominantly that is kind of co-occurring in Asia, like China with around 109 million incidences, India with around 70 million incidences, as well as US with around 29.3 million incidences. And looking at that, it is really critical that we have to really look into our lifestyle. Plus, also we need to focus a bit more on dietary preferences. And it is clear that one of the remedies what we really need to think through is like how do we take a look at some of the staples, including rice, where that has been really the bread and butter of of South Asian populations. And so when we really looked at many of the released popular varieties of rice, we noticed that the glycemic index of all the rices have been extremely high, ranging between 70 to almost 94. So the glycemic index is measured in terms of, when we consume, let's say, glucose, and when we measure glucose concentrations in our blood, that will be treated as 100% in relation to that. Any given variety where a glycemic index is going to be high means the starch is rapidly digesting into glucose, and that gets dumped into a bloodstream. So given that scenario, it's really important that we have to make some systematic effort to lower the glycemic index of these important or popular rice varieties. And so keeping that in mind, basically, we have been doing some specific efforts to kind of manipulate the glycemic index of rice. So at first, let's take a look at how the selections have happened between various grasses. If you see the availability of carbohydrates, the brachypodium, which really had less available carbohydrates, but over a period of time, we have been making breeding varieties and making selections where available carbohydrates have been increased. And rice is one of the crop where available carbohydrates have been extremely high. That has been done by changing the composition of grain, where we have been increasing the percentage of starch content in the grains, and we have been reducing the dietary fiber source as well as protein. And other components includes basically lipids and other major storage products. And clearly what we have been really doing is we have been targeting to increase starch content in the grain to increase the availability of carbohydrates. And as a result, many of the rices what we do have, the carbohydrate content, we are consuming with around 225 to 310 grams. And even though there is enough carbohydrates which can actually provide energy through the carbohydrates, we can meet nearly 50% of the protein requirement. And interestingly, there are many essential micronutrients that does not cover as part of the staple diet. So as part of this strategy, one simple shift could be that we could really look into kind of avoiding the processing. So instead, we consume the whole grain of rice, which is nothing but brown rice. So as a result, that we could try actually to kind of have the possibility to include enough fiber. Like if you compare the milled rice, which has only around 0.4 grams of fiber versus 3.5 grams, that's a good intervention. Likewise, various vitamins, you clearly see that like B3, B5, as well as folate like B9, there is a very high percentage if you would continue to consume basically the whole grain consumption. Likewise, some of the minerals are also present at abundant levels, which can provide the daily requirement of these minerals and nutrients. But quite often, we cannot store the brown rice for a very long time. There is a rancidity problem, and also consumers, because of texture and palatability issues, they're not consuming the whole grain. So as a result, we had to do quite a bit of an effort to make sure that besides providing the calories, we also have to work around to bring the missing nutrition back into the mill rice. So, and here is an example where across the globe, basically we are, providing the food through staples like rice, wheat, maize, and potato. All of them, they do have very high glycemic index, which has been reflected here. The popular varieties in rice, the glycemic index ranging between 68 to 94, whereas in wheat from 70 to 90, maize between 60 to 80%, and potato 90%. So this is where we really need to concentrate about the quality of starch instead of just the quantity of starch. And also when we really look at the plates around different target countries, we see either we have been consuming really the highly processed foods or even energy drinks. And even in terms of different target countries, we see that the proportion of rice what we are consuming is very high. Again, not all community members could actually afford the diversification, and this is where we really need to look into some of the tangible solutions to see how we can bring the missing nutrition back. So at IRI, we have collected more than 130,000 diversity of germplasm across the globe, and that has been stored in our gene bank. For decades together, that germplasm has been sitting in the cold storage rooms in the gene bank. And nearly 7 years ago, we have launched the program, and we have brought in various enabling technologies to tap in the diversity of gene bank. And that's where we have really created a huge big data sets that we have sequenced nearly 5,000 genomes of rice, and we have been phenotyping, nearly around 15,000 various accessions of rice. And using that genome-phenome linkages and bringing in basically various artificial intelligence tools and techniques, as well as post-genomic technologies, we have been undertaking the gene discovery to see whether important alleles and haplotypes could be discovered in those land races. And so once we were able to identify the donors from the gene bank for nutritional intervention and having a discovery program put in place, then we have taken up basically a precision breeding approaches to bring missing nutrition into rice. So the approach what we have actually undertaken is quality. Breeding, and that is where breeding for diversified nutrition and positioning those varieties to cater the requirement of market segments is really an important target. And so as part of this nutrition, what we have been really looking through is we have been focusing on important triad like lowering the glycemic index, also enhancing the dietary fiber, then looking into enriching protein with essential amino acids, wherever appropriate, we are looking through for enriching antioxidants or vitamins, and of course, micronutrient dense rice. When we do all of that, it's also equally important really to look into food safety issues, because there are many target regions in various countries. We do have heavy contaminations with heavy metals, and also pesticide usage has been tremendously increased. And as a result, if we are able to breed varieties, then that has to ensure the food safety aspects. So while we kind of looked into all the gene discovery program, we have come up with the strategy. On the left side here, we see that many of the rice varieties in the milled rice, it doesn't have any dietary fiber, just below 0.5%. Starch is around 90%, and protein is in the range between 6 to 8%. So what we have really done is we have optimized actually the strategy, keeping the starch around 90%, but we have changed the amylose to amylopectin ratio in such a way that we can change the digestibility of rice. As a result, the low glycemic index property can be brought in. And here we have targeted actually the degree of polymers of amylose, reaching up to 15% of amylose 1 target, and 10% of the target of amylose 2 fractions. And that resulted in lowering the glycemic index. On the other hand, we also have been developing some of the rice varieties to increase about 10% of the protein content. And we are also adding flavonoids or anthocyanins in specific distinct product segments of rice varieties. And of course, rice is also being positioned for processing food products like pasta, noodles, as well as various cookies and functional foods. And so it's really important to explore to basically add the dietary fiber source, and we have been strategizing to increase ranging between 5 to 9% of the target. So I would like to kind of give you a couple of examples of what our learnings are when we screen the gene bank varieties. So as I told you that when we looked into the glycemic index property of many popular rice varieties, they were all very high, ranging between 70 to 94. And for the real intervention, a GI of a particular food product should be below 55. And so we have started actually to screen the germplasm of gene bank material. And interestingly, there was a notion suggesting that the GI is inversely proportional to amylose content. But interestingly, when we really screened the germplasm and looked into that dynamic relationship, the correlations were very, very minor. It's only 0.28%. Of course, low amylose types or waxy rices, they had very high GI. While we tend to bring it to the intermediate levels, still it remained from very high to high. But interestingly, within the high amylose types, what we noticed is the GI spans from very high to low. So this is where I think we have been really fixing many of the popular varieties, particularly in India, Bangladesh, Pakistan, Sri Lanka, and Nepal, with intermediate to high amylose types, mostly between 24 to 26. And as you could see here, the GI varies a lot from high to almost low. And so amylose could not be used as a proxy measure for selections of lowering the GI. And so this in vitro method of screening for the glycemic index was rather important. And we did identify 3 low GI donors out of that screening, which really had a GI of 55 or below. And then through AI and post-genomics pipeline, we were able to identify the genetic basis of the glycemic index. We identified 2 different chromosome regions, chromosome 6 and 7. And then we did identify important haplotypes to basically undertake a pre-breeding approach. And the donors, what we have identified as a low GI index rices, were fed to the human subjects, and we were able to confirm that the GI values of those donor lines are 50 and 51.1, and the blue line shows that that's a glucose standard. And so we then also kind of conducted the sensory panel assays of those low GI lines, and we compared that with high quality rice. So here is an example like Dinorado. It's a popular variety used in Philippines. And when consumers, they eat this particular rice, then they describe the texture of this rice being very high stickiness to lips, high stickiness between the grains, and it has a uniformity of bite with high initial starchy coating. And interestingly, one of the low GI lines, what we have identified, matches the textural attribute of initial starchy coating, and it has intermediate property for the stickiness of the grains. Whereas the other two low GI lines, basically the initial starchy coating is way too low. And so based on this, we have identified that this particular donor is actually rather suitable to breed a variety with low GI property, matching the texture of Southeast Asian population, whereas the other two lines are preferred by basically South Asian population. So that's the kind of approach what we have taken, and utilizing that massive amount of data what we have generated between genome phenome analysis. Here is an example where transcriptome-wide association studies were done, and we have identified a genetic region of chromosome 6, chromosome 11, as well as a few SNPs on chromosome 5. And here within this chromosome 6, we identified basically more than 50 different genes having a stronger marker trait associations. And interestingly, when we used transcriptome-wide association studies to narrow down the target, we identified basically a cis effect where some of these particular high-value SNPs, what we identified, we were able to narrow it down to 21 target genes. And the transcriptional regulation of these genes, they are under basically cis effect. And so we were kind of interested to dig into that mechanism. And before we did that, we tried to identify some high-value SNPs. So these are all the high-value SNPs what we have identified within the key gene called granule-bound starch synthase. And basically, we were able to identify an important SNP. One is which is sitting in the exon-intron boundary, influencing the alternative splicing, whereas the other one basically in the exon 10. Just with these two SNPs, we can really convert a variety from very high to intermediate GI, transitioning from T to G SNP variation. And when we really have the second SNP C and T sitting in this exon 10, basically we saw that led to the differences in the texture. A variety with having a GT would be hard and retrograded in its texture, whereas a haplotype with GC would have a soft texture, but still have that intermediate GI. So these are the natural SNPs what we have seen that different populations in South Asia and Southeast Asia have been selecting without the prior knowledge of those SNPs or genomics knowledge. And basically what we have shown is the variation, whatever was existing in rice, can only explain from very high to intermediate. But if we really need to move towards lowering the GI, we need to tap in other candidate genes sitting in this hotspot recombination site. So here is actually a set of 21 genes what we have identified for the cis effect. And interestingly, what we noticed is all of that was under the influence of methylation. So we clearly noticed that some important genes like 4230, 4240, and 4070, they were subjected to kind of, when it was fully methylated, then that had a relationship to the GI. And so easily we could actually target those important target genes, and then we could modify basically the glycemic index of a rice variety without losing texture. And so by deploying that knowledge and by looking into gene regulatory networks, we could clearly identify that, besides that amylose amylopectin variation, there are other important genes sitting next to that genetic region influence phenylprepanoids as well as some of the dietary fiber, you know, in the milled rices, which plays a vital role in lowering the glycemic index. And so we rather concluded that such a, like a systems genetics approach, what has been deployed rather helps us actually to identify high value important genes sitting in that hotspot region. And we did define actually the top candidate genes and define basically the haplotypes, and this information we have been deploying it in the pre-breeding approach to develop low GI rice varieties. And in parallel, we also took the strategy to go into the genome editing. And this is where we have been looking into transcriptional and post-transcriptional regulation. Here is an example of granule bound starch synthase, which has been considered to alter not only the amylose content, and that also had the influence on the glycemic index value. So this particular strategy has led to develop various rices with haplotypes. We call it as a VAXI-A, VAXI-B, VAXI-LB. So just by looking into that variation, we were able to create different cocktails of taste and textural preferences. And considering that gene plus other target genes, what I've showed you, we have now actually lowering the GI by taking into consideration the taste and textural preferences. And here we have undertaken precision genome editing technologies to develop additional rice varieties. And this is an example where we have identified additional target regions for the resistant starch, where there are certain accessions with moderate resistant starch versus low resistant starch has been identified. And this is another known gene of starch synthase 2A, which was identified to be critical. And another important gene, downstream, we did identify that as another critical factor to enhance the resistant starch in rice grains. And deploying all that knowledge, we were successfully able to move many of the iri varieties which had very high GI towards basically the low GI. And here you could clearly see that lines below 55 GI has been generated with a pre-breeding approach. And on the other hand, we also looked into the possibility whether we could develop an ultra-low GI rice varieties in high-yielding background. And here we were really able to develop ultra-low GI rice varieties with a median of around 30. And if you see other rice varieties, they would really range between 80 and up. So this is a real population what we have created. We have deployed certain specific combinations of haplotype. Just with around 10 SNP variations, we were able to convert basically a very high GI to an ultra low GI. And also we were able to overcome the yield barrier by changing the panicle architecture of the rice varieties. And so currently these lines have been fixed, and we are testing for the observational yield trials and advanced yield trials. And eventually these lines will be available for making specific food products in near future. So that's one example what I really wanted to point out in terms of emphasize the importance of post-genomics technologies, how we could deploy into the breeding, where we could really develop, the low or ultra low glycemic index rice varieties. And we also have been looking into another example where in the endosperm that we wanted really to enrich a molecule called catechin, which had the anti-cancerous properties. And here we did identify another region on chromosome 7. So this particular target gene is responsible on one hand for lowering the GI. This is sitting on chromosome 7, and clearly you could see that unmilled rice, it would really have extremely low GI. And likewise, we also see the median of milled rices, in an acceptable range. And interestingly, the total flavonoids have been extremely high in those particular rice varieties. And so when we basically did, the anti-cancer experiments, particularly using cancer cell lines, we noticed that lines with this particular haplotype 4, they really exhibit higher cytotoxicity assay as to the zeosin controls. And that was true for colon cancer, liver cancer, breast cancer. And interestingly, now what we have been doing is like we are repositioning these particular lines, that even with milled rice, that such rices enriched with catechin content could be utilized as source of functional food. So, and then meanwhile, we also have developed, with the help of Government of India, an overarching framework. Particularly, the concept of foodomics has been put in place, where we are linking all the way from developing nutritious varieties, testing basically the byproducts, the bran and other broken flour, to really come up with functional foods. And those could be used as supplements. And at the same time, where the strategies what we have been putting to enrich nutrition in the milled rice is continue to promote as part of a milled rice consumption to translate human health benefits. And so I would like to go back to this concept. At the moment, I showed you that we do not have enough nutritional diversity in staple, but we have been making so many distinct products in the market. So if you see that baby food for pregnant women, even for the adults, various breakfast items, we have been processing varieties for coming up with distinct food products. And when we further process those varieties, then the glycemic index would be way too high. And many of these varieties do not have the required nutrition. So that is part of this problem what we are creating, where because of demand, we have been coming up with highly processed food products. And this is where we really like to propose that, taking up those specific breeding programs to bring in the missing nutrition and reposition these nutritious rice varieties will pay, a big impact, because already the value chains have been established. The food industry has been around to make these products, and we really need to develop those linkages and demonstrate those linkages. So that is where, as part of one CGIAR initiatives, IRI has been putting in place to streamline the breeding of nutritious rice varieties, where we have really designed the concepts, and we have come up with the breeding strategy optimization. We have been developing the product concepts, and those varieties have been tested at a stringency to look for those nutritional triads, plus ensuring the consumer acceptance criteria, and finally looking into positioning those nutritious varieties for health benefits. So that pipeline we have put in place, and we have implemented that to kind of streamline the nutrition program at IRI. And also in India, with the help of Government of India, we have established basically, the entire food quality testing lab and product development lab. And we also have established a lot of analytical platforms over there to really test a lot of nutritional attributes. And we have been prototyping various food products using those nutritious rice varieties what we have been developing, so that we could really create a good alternative to the current challenges. And so at the moment, we are also negotiating and in discussion with various state governments. And here we have come up with a strategy to put in an incubation center where we have a technologies placed almost from innovation of development for the varieties to food product development, creating basically a scalable technology production, bringing in the industry and government stakeholders, and then develop good market assistance, and then properly guide them to to take it forward in terms of creating the needed impact. And for that, we have come up with the strategies from concept to launch of those food products, where particularly we wanted to empower women entrepreneurs for the value addition. And we have put in, good strategies around with various state and central governments in India. And also we have been working with APEDA, who are keen in leveraging the value for this, and making sure that we not only just export low quality rice, but we really attract high premium and really create a good leverage of value addition to our rice farmers. So going back, actually, we are used to thinking of really nutrition through diversification, where if we really tap into the existing diversity, we could capture enough nutritional requirements. But quite often the problem is perishability of those products, and lack of good value chain around those products makes it very difficult. And also low income community members, they cannot afford to have nutritionally rich food products on the table all the time. So for that purpose, we have put in place a two-layered mechanism. So one is really all the way tapping the diversity through modern technologies, bringing in the pre-breeding or precision breeding approach, and then really develop nutrient-dense varieties with optimum calories. And eventually, once we have that and deploy that as part of a food product or the whole grain or milled rice, then we also have to really test the health benefits with the biomedical community, where we really need to customize the healthy plate, by looking into the nutritional density versus calories. And also when we really feed in, we have to take into consideration the gut microbiota and monitor basically human health biomarkers. And so thereby we could clearly come up with more generalized nutrition solution, or also we can come back with personalized nutrition strategies to really take that as part of an intervention in future. So what I really like to kind of highlight here is, I hope I was able to kind of show you the strategy that in breeding, we were able to create low GIR, ultra low GIR rice varieties, where we have developed high fiber related varieties, which helps to prevent colorectal cancer, improve healthy bowel function. And in rice with high protein, having no allergenic proteins helps to cater the requirement and prevent celiac diseases, especially for the gluten intolerance people. And of course, low glycemic index alone itself can actually contribute to reduce diabetes incidences and prevent non-communicable diseases. And also, IRI has been working to enrich zinc through mainstreaming zinc in many programs. So we're right now we are able to increase 20 to 24 ppm of zinc And also through transgenic technology, IRI has developed golden rice to enrich vitamin A related lines. And Philippines has released those, golden rice varieties, and currently they are being tested. And additional technology what we have developed is really fortification for folic acid or for enriching iron, where we could not do that through breeding. And interestingly, here in this fortification technology, we have developed ultrasonication techniques where we are able to enrich the concentration. Like if you see here, the concentration of folic acid, we were able to bring it to 165 milligrams per kilograms. And so just, 2.4 grams of these fortified rices could be mixed per kilogram of rice. So this is where we are again discussing with Government of India to link some of these things to midday meals and other programs where such fortification technologies for enriching folic acid or iron could be utilized as part of an intervention strategy. And on the other hand, where we are able to bring success through breeding or through genome editing technologies, then we have created series of health and nutritional value rice varieties. And we also have been creating a pipeline to develop various product prototypes and take that scalable technology to demonstrate to the industry and government sector so that the entire value addition can be captured hand in hand. So going back to my introduction, basically when we do have a healthy plate, we really need to think of vegetables, fruits, whole grains, so on. So that actually gives the source for complex carbohydrates, essential amino acids with proteins, soluble and insoluble dietary fibers with micronutrients, vitamins, and of course antioxidants. And interestingly, with the kind of varieties what we have been developing, it gives an option where consumers can pick actually for a particular type of meal, having either pigmented rice or low GI rice or high protein rice, and particularly including those germinating sprouts. That can be really a good alternative to meat protein. And so that could give additional sustainable interventions in near future. So to conclude, basically future health of humans that depends on a simple formula, nutrient density over calories. And so that future health is really determined based on that nutrient density per calorie in our healthy plate, what we put in. And of course, we have to continue to put efforts to really bring in source of antioxidants, micronutrients, vitamins, and dietary fiber, either through diversification or through those specialty staples what we are creating. And of course, once we create a good value chain around and food products what we develop, which are enriched with complex carbohydrates with all those essential nutrients, that definitely will help to translate nutritional security targets in the given country. So going back to our mission of IRI, so healthier rice, what we have been developing is going to provide a sustainability. And this rice system approach with healthier rice can actually improve nutritional security in future. And also it can bring sustainability. And that's really, really important factor, because all these varieties we could, whatever we have developed, we could store it like any other regular rice. And perishability has been really overcome by bringing in the nutrition. And so in order really to translate that, we need partnership, particularly with biomedical community, so that we could really bring in and generate enough data to show these health benefits, and then we could continue to work with various government stakeholders to add value to the consumers and of course bring in premium to the farmers. So I just would like to give you one example. A normal rice variety at the moment, it fetches only around 250 US dollars per metric ton. Whereas a rice variety with low GI, because of the demand, it can fetch up to 1700 US dollars. So if farmer has to just think about high yielding variety, they won't really get that kind of a higher premium. But if we are able to bring in varieties which can fetch higher revenues, and when we ensure that they are high yielding, then that can actually bring not only the rice efficiency, but also it would help address the UN sustainable goals and improve livelihood and health of the farmers. So with that, I would like to kind of close my talk, and I would be happy to address any open questions from the audiences. Thank you so much, Dr. Kavakishore, for this opportunity.
Dr. Srinivaslu, thank you very much for a wonderful talk. Okay, you have few questions now. Are you able to see the audience here? No, right?
No.
It's fine then. Okay, I'll just.
Good morning, sir. I am Indrani and I'm a final year PhD scholar. I'm pursuing in the field of cancer biology and drug research. So I have a couple of questions, sir. In one of your presentation, you mentioned like you have performed haplotype mapping and then you have checked with the anti-cancer properties in different cancer cell lines. So I just wanted to know, did you do any characterization studies on along with the haplotype mapping, sir?
Yeah, so as I kind of showed briefly in my presentation, what we did was we have first discovered the genes responsible for enriching catechin content as well as the total flavonoids in the milled rice endosperm. And also we characterized that elevated proportion was even higher in the rice bran. So once we were able to figure out the candidate genes and haplotypes, then we have identified the lines possessing that superior haplotype versus inferior haplotype. And then we have actually subjected basically bran as well as milled rice for the cytotoxicity assay using those cancer cell lines. And basically, we conducted time series experiments along with the controls, and we very clearly noticed that the haplotype 4 possessing lines did exhibit higher cancerous property. So at the moment, we are actually went ahead with purifying those molecules, and we have been preparing the scalable production for catechin isolations and preparing basically the supplements using the rice bran, which could be positioned for nutraceutical supply in the future.
Thank you, sir. You have answered my all questions. Thank you, sir.
Any more questions?
A wonderful talk, sir. This is Prash. Sir, there could be, I mean, while jotting down to the risk alleles or the allele-specific traits here with respect to the low GI, how could you evade this linkage disequilibrium, sir? Is there any way that you could probably get into this? Yeah.
Yep. Thanks. I think that's really a very, very good question, Dr. Prashant. So, as I told you, like that region was basically, already a fine mapped region. But interestingly, what we noticed is there is a very high rate of recombination happening around that region. So even what we noticed is within granule bound starch synthase, we could actually see that recombination happening for specific types of haplotypes. So what we had to do is like really create a very high density mapping populations and basically go through and screen for the haplotypes and then pick the superior lines where the right recombinations happened in those lines. And once we pick those superior haplotypes, then we subjected that to in vitro GI analysis. And then eventually we then moved to the glucose sensors and in vivo assays on Caucasian population, Filipino subjects, and currently we are testing on the Indian subjects too.
Wonderful, sir. It was an absolute scientific treat. Thank you so much. Yeah.
So, yeah, we have one more question.
Sir, it was an excellent talk, and my question is going to be a general one. Has there been any CRISPR type intervention for reducing GI?
Yeah, thank you. I think I showed you in one of my slides, particularly we have been using CRISPR-Cas9 genome editing strategy. And as a proof of concept, we have been playing around only with the granule-bound star synthase gene, and we have created different rice varieties with various textural preferences based on the waxy haplotypes. And once we have done and standardized all the technology and created different types of rice with different texture, targeting the GBSS, additional genes which was sitting next to that, which I showed, is also critical to moving the GI to low from intermediate or high. That we have been now focusing on to develop genome edited lines. And so in near future, then we would really combine those particular targets coming downstream of GBSS target genes along with the GBSS haplotypes to take care of taste and textural preferences. So given that recently Government of India has deregulated, I think that's where we are now accelerated, as part of this precision breeding strategy to see whether we could really target the specific haplotypes what we are interested in to edit through CRISPR-Cas9 technology.
Sir, I have a further question on this. When you're trying to make intervention through CRISPR techniques, there must be minimum change in the fluxes of the other components. So how do you manage that? Yeah?
I couldn't really capture your exact question. Can you repeat?
So, see, because of this editing, there can be the flux changes in other components, changes in fluxes, yeah.
You mean biochemical fluxes?
Right, yeah.
Okay. No, I think the targets are very, very precise. So basically, as I said, when we were targeting granule-bound star synthase, editing happened only for few specific nucleotides. And that change is pertained only to basically starch composition. And when we are targeting additional candidate genes sitting downstream of the targets, then those targets are more intended for polyphenolic compounds as well as for dietary fiber, nothing else. So I think this precision editing strategy is actually a more appropriate method where we will not expect any side effects around that. And in our haplotype breeding strategy, of course, we we have been more careful to really increase recombination and select actually the right haplotypes. Once we have identified the right haplotypes, confirmed the phenotypes and all, then we have taken up a lot of backcrossing strategies from the RILs to make it near isogenic lines, and then those near isogenic lines will be the one where we will be using as the final breeding product.
Thank you, sir.
Okay, just there are no more questions from audience. I just would like to comment. It's really an excellent and exciting talk, Dr. Srinivasalu. I appreciate your efforts and not only generating different traits with nutrient values, importantly that anti-cancerous property. Okay, I see actually, I'm from Hyderabad and I see this Asian Institute of Gastroenterology. You know, if you go there, that hospital is full of patients with, different diseases and all, especially related to gut problems. Your contributions are very important. And today, I mean, hearing you, the work trying to improve this kind of rice varieties using nutrigenomics is very significant, and I greatly appreciate. But I have a very small question, like not only the nutrient values and also this anti-cancerous properties of the grains. You can see this japonica, this Japanese rice, you may know about it very well. It's very tasty, though it is sticky. So it's probably, if I'm correct, it is about 25 to 25% of amylopectin in that. Is that the reason? Maybe that is very tasty. So are you working on such lines also to develop such traits also?
Yep. So actually, Japonica rices, particularly if you're talking of jasmine type of rice or
Jasmine.
Kaudatmali or Koshikari,
Mm.
these are very, very popular Japonica rice varieties known to have very unique aroma and soft texture.
Yeah.
So most of those varieties were bred with very low amylose. That means it is enriched for amylopectin. So those varieties, amylopectin composition ranges almost from around 85 to 90% of amylopectin,
Okay.
and because of that, it attains actually that soft texture. But of course, it also turned out that many of those rice varieties were found to be very high in its GI. So, for instance, the Kodak Mali as well as Koshikari, the glycemic index values surpasses 96. So that's a bit of an unfortunate scenario. So yeah, I think the solution would be, like as I said, we really have to strike the balance between taste and texture versus nutrition. And also we need to be very conscious about the amount what we eat. So that glycemic load is another important factor which we really have to take into consideration in our healthy plates.
Yeah.
So again, in general, I really like to kind of make a request to the biomedical community in India. So if we can come together and work in a synergistic way, we will be very interested to really collaborate, and particularly we need to create a lot more evidences on those nutritious rice varieties in terms of systematic interventions. And once we have that kind of a data, then I think we could speak more confidently with the policymakers. So we still have quite a bit of a way to go. And it's really high time that as a community, we need to think together and come together and work together on that important frontier area. Thank you.
Oh, thank you very much. And so I just would like to thank both the speakers. The first speaker, Professor Verma, and also Dr. Srinivasalu, for your excellent talks today. It's really a big applause. And really, it's a scientific feast, and I'm really excited about this. And I thank you very much, Dr. Srinivasalu, on my personal behalf and also on the behalf of organizers, Professor Kavikishore. I thank also giving me this opportunity to chair this session. Thank both the speakers. Now.
Thank you, sir. As sir mentioned, it was a wonderful scientific session. And we also thank Prakash sir for chairing this session. And I also thank the rapporteurs. Now we will take a short tea break and come back for the next technical We have 3 lectures by Dr. Pushpendra Singh, Dr. Jeeban Jyoti, and Dr. Bipin G. Nair. And this session will be chaired by Dr. Krishnamohan Medicherla from BIST Jaipur and Professor Ratnagiri Polavargaru from Genomics Biotech. May I request the chairpersons to please take over the session, and the rapporteurs for this session are Dr. Sampath Kumar and Dr. M. Indra.
Yeah, welcome back to the technical session 4. We have 3 lectures today in this session. Without wasting much time, I would like to request Dr. Pushpendra Singh to start his talk.
Thank you very much. I thank the organizers of this conference, especially Dr. Kavi Kishore sir and the entire team, Prash, and everyone who has invited me to be able to share with this work with you all. So, the title of the, I'm currently working as a scientist E at ICMR, National Institute of Research in Tribal Health in Jabalpur. It's very much close to the heart of India, geographically the center of India actually. My name is Dr. Pushpendra Singh, and I have joined ICMR around 3 years back. And the title of the work that I am presenting is Comparative Microbial Genomics and the Transmission of Leprosy. Maybe if I stand there, is it? It was working actually, so if it is helpful, I can. Yes, it is working, I think, no? Yes. Okay, thank you very much. So, leprosy is an ancient disease, very well known to humanity and had visited almost every civilization. And even though people think that it is a disease of past, nearly quarter of a million new cases are reported, and you can imagine that many more go unreported also. Of these, nearly 60% are reported in India, which is a disproportionately high burden of leprosy in India. It's an uncultivable pathogen, and its transmission mechanisms are not fully understood, though the most commonly believed mechanism is through aerosol transmission via droplet infection. Its genotyping scheme is very difficult because there is almost no variations between strains from a closed geographical area. And there are complex host genetic factors which make the leprosy susceptibility a very difficult aspect to predict. So nearly 95% of the global population would not develop disease even after they are contracting the bacillus, but the remaining 5% can develop different type of disease spectrum. So the causative agent of leprosy is Mycobacterium leprae, but there is also one more that we will hear about in this talk. That's Mycobacterium lepromatosis that was reported by our group a few years back. So the genome size has already reductively evolved from nearly 4 million to around 3 million bases. So there is a nearly 25% reduction in the genome size, and over the course of evolution, it has accumulated large number of pseudogenes, which means that it has lost a lot of metabolic and catabolic potential actually, and that has actually made it host dependent, and there is extremely low level of genetic diversity. So there have been some animal reservoirs reported in last just 1 decade actually. 9-banded armadillos is known to have natural infection of leprosy, but it was not known whether they are, the strains are same or not. So we will hear a small story in this presentation about this aspect, but more recently in Europe, there were reports of these red squirrels carrying natural infection of leprosy. In a country where the disease has been eradicated since last 6 or 7 centuries, if you find some animal carrying this without being detected for this much of advancement of technology, this is very surprising. And more recently, just last year, in these chimpanzees also, there have been the strains of Mycobacterium leprae reported. So the striking thing about the red squirrels was that not only one species of leprosy bacilli, but both the species of leprosy bacilli, Mycobacterium leprae and lepromatosis, were actually identified in, in these animals. So are there any animal reservoirs in India? We don't know that yet, but I think systematic investigations are also not being carried out so far in this area. So as I mentioned that the genotyping is very challenging because the bacillus has very few variable positions. So if you classify based on just a combination of 3 SNPs, we can put them into one of the 4 genotypes, 1, 2, 3, and 4, which can be further subtyped based on few more positions. And what this genotyping scheme tells us about the human migration, and it has actually, it was earlier widely believed that leprosy, because it was very well reported in Indian texts, in Sushruta Samhita 600 before Christ actually, before Common Era. So people used to believe that it was originated in India, and with the Sikandar's army, it went to, or the Alexander's army, it went to Europe. But the genotyping data doesn't actually show that, because the strains present in India are genotype 1, and the one which are present in Europe are type 3. And the more logical route is that it originated somewhere in the Africa or Central Asia, and then with colonization, it went to Europe via Silk Road, and then via colonization it was introduced into the New World in Americas, and type 4 originated independently. So basically by genotyping we can also find this type of long-held beliefs, whether they are true or not. So you have recently all are aware that ancient DNA work of Neanderthals has got Nobel Prize. From the group of Svante Pabo, Dr. Johannes Krauss was a postdoc with which we were collaborating, and we carried out the investigation of the leprosy skeletons from the medieval Europe. And there we showed that the strains which were present in skeleton samples were almost identical with those present in the currently infected armadillos in Europe. That basically shows that there was a link of maybe most likely through the colonization itself. So our primary question was that how the strains of M. leprae in medieval Europe looked like, whether they were less virulent, that is why it eradicated by itself without any drug, without any vaccine in 13th to 14th century. But that was probably most certainly not due to the loss of virulence or different type of strains. It was probably other host genetic or demographic factors. For example, the plague might have a disproportionate toll on the leprosy patients. But other investigation that we wanted to know was whether it is the only species that is associated with leprosy. And we identified that certain patients have a different clinical presentation known as diffuse lepromatous leprosy. And in those we sequenced a new species, Mycobacterium lepromatosis, which has been evolutionarily different from Mycobacterium leprae for more than 14 million years But despite that, and despite being known to humans for last 150 years, it was believed to be the same organism. But we found that this is a entirely different species, though evolutionarily very much similar to Mycobacterium leprae, but also very far in a scale of 14 million years. So what does this genotyping tell us about the animal reservoir in Americas? It is known from 1975 that these animals, armadillos, 9-banded armadillos particularly, carry natural infection of leprosy, but and it has been actually used as a resource for leprosy research for the availability of viable bacilli, because Mycobacterium leprae is not a culturable organism. And there has been a lot of human armadillo interaction for several type of souvenirs or as a pet, like you can see here, even as a food source, or these are also known as the Texas speed bumps in Louisiana or southern part of US. But when we sequence them at the whole genome level and compared the strains derived from the humans living in those areas where naturally infected armadillos are also present, we found that they were almost identical at the genome level. That was the strong evidence that proved that it's a zoonotic leprosy in southern United States. The editor insisted to keep the word probable, but later on in Florida also we proved the same study, and in the subsequent paper in the emerging infectious diseases, this probable word was not required. So we just discussed about the history of leprosy in Europe, and this was from different parts of the Europe in 3 countries. So what I was trying to emphasize here is that the ancient DNA is very much fragmented, and also there is a lot of human DNA contamination and the contamination of a lot of other microbes which decompose the body. Despite that, we were able to get the whole genome sequence of the strains which were 1,000 years ago present in this medieval Europe. And that was possible because of the positive capture on an array or by removal of the human DNA. And I think this can be applicable for several other pathogens or for the specimens where the culture is not possible, or due to some regions related to biohazard, it is not doable. So using that, we have compared that this strain from the USA and the Europe were almost identical at the genome level, just 28 differences in the 3.27 million bases of the genome. So the important outcomes of these genotyping studies are shown here, that natural infection of leprosy is also in relation with the zoonosis, and the strains in America reached Europe via colonization from Europe, and lepromatosis is a new species that has the same level of neuropathogenetic potential, because the genome is almost identical, though they have been evolutionarily very different. And the disease agents can remain present in wild animals for centuries undetected. For example, M. leprae in armadillos, red squirrels, and chimpanzees. So we had compared the genome level also. I'm not going into those details here. Our group has recently also carried out the whole genome comparisons of M. leprae strains, and there we found that most of the strains actually belong to genotype 1D in India. So it is not possible to genome sequence all the strains because of the cost. So we thought that, and I'm zooming out to the strains which are of genotype 1D, which is present in India. And why not actually think of some innovative ideas that we don't need to actually sequence the whole genome. And for that, we compared the strains of genotype 1D and found that some SNPs are unique. So targeting those SNPs by restriction fragment length polymorphism, PCR-RFLP, we can make it much more simpler to identify the strain and their genotype without having to even sequence it. So if it is a genotype 1D, it would give this type of band, and if it is subjected to restriction fragment analysis, it would be giving 2 bands of 100 bases and 72 bases from a 172 base pair product. So this is basically very useful approach. Similarly, another prominent genotype can be identified based on the even base pair repeat that is present, which gives a higher base pair size of this strain. In addition, there is no point of care test available. So we thought of using these isothermal amplification assays. By visual detection, we can detect the strain up to 95 bacilli per reaction. But on electrophoresis, it can be up to 9.5 bacilli also, because we have targeted some multi-copy repeats which were identified by the whole genome comparison. And these LAMP assays have their own problems because they can be non-specific. So again, the LAMP product can be subjected to PCR RFLP, and that would be much more convincing evidence that the amplification was specific. In addition, we have been talking about the CRISPR-Cas based method, and since there is currently no point of care test available, and gel electrophoresis is also not a field friendly method actually, we have tried to adapt this method using CRISPR-Cas and hybrid detect lateral flow strips. So we can see that the positive sample gives a test line and the negative sample gives the control line. There is some visibility on the test line also, but this is actually very clear that one can easily identify the signal even in the field without having to have any real-time PCR or any type of PCR, because RPA is isothermal amplification that can be performed at 37 degrees Celsius, and it can be performed by any exothermic reaction or even by the body temperature. So the summary of work is that we have developed some cost-effective methods for whole genome sequencing of uncultivable pathogens, which can be not only applicable for leprosy but for other diseases. Comparative genomic analysis have revealed some informative loci which can be directly identifiable for identifying the strains prevalent in a particular area. That can allow simultaneous detection, identification of the species, and genotyping. And that can be actually adaptable for the drug resistance studies also. So far we have not actually gone to that level. And simple field-friendly test is under development for POCT. And I thank the entire team, which has actually been very helpful, especially Mukul and Purna. Mukul is a postdoc and Purna is doing her PhD. And the funding agencies from different sources are shown here. And I thank you for your attention. And any questions are welcome.
Thank you, Dr. Puspendra Singh. The floor is open for discussion. Couple of questions. And I must say that I used to think the paleogenomics is always bothered about the bacterial DNA contamination. But now I see that when you are looking for this kind of strains, even the human DNA is of a problem.
Yeah, so my question was, you said you biotinylate the human DNA. How do you prevent biotinylation of bacterial DNA at the same time?
So we prepare the biotinylated library of the pure human DNA separately. Okay. So that is used as a bait. Control. And on a streptavidin bead, we remove it.
That's used as a base?
Yes, so a bait library is used. Yes. Which is on a different type of sequencing system. It would not get sequenced so on the those, subtractive hybridization basically. And that actually allowed us to the genome sequence the new species of leprosy. I think that can be adaptable for the Helicobacter pylori.
In fact, science should be taken to society. This is what we have been deliberating for the past 1 day or 1.5 days. How do you think, what is the current scenario of the problem of leprosy in our country, and what do you think that the ongoing efforts would be able to address the current problem of the leprosy in the country?
So it was declared as a disease eliminated from India in 2005. That sent a different type of message to political people. They said that, okay, leprosy is eliminated, let's focus on other disease. Let's put the staff involved in the leprosy control activities into other programs, into filaria, into TB, and this basically has been a it has backfired actually, and that in the process the expertise has been lost, and there are even very big challenges in training the new generation actually. So I think the focus has to be not on elimination but on the increased notification of the disease. Higher you detect, lower is the transmission, and that is how basically translates into the later on control of the disease or reduction. We shouldn't be actually claiming that we have eliminated a disease too early actually. I think that is where I think focus should be again, because a lot of new cases are being reported in several states. In fact, 11 states have started showing the rise of leprosy. So that is something worrisome. Yes, sir. I actually wanted to come to that. The tribal population represents only 8.6% of the Indian population, but nearly 20% of the cases of leprosy, newly identified leprosy cases, of that 20% are among the tribals. So that is a disproportionately higher because of the remoteness of those people, or lack of awareness, or they are not actually, their health seeking behavior is also not as per the norm actually. They would seek the traditional healers first.
Right, right. So,
Any, please.
So which is a particular organ or a tissue where this leprosy resides inside our system, sir?
Thank you very much, sir. This is a very important aspect of the leprosy biology itself. Leprosy has, during the reductive evolution, it has lost the heat shock proteins. So it likes the low body temperature. So the extremities of the body, like hands or the nasal mucosa, because the in the initial stages, it is reported in the extremities of the body. It can affect the skin, but mainly it targets peripheral nerves. That is how it leads to the loss of sensation, and that is how the people who have leprosy in advanced stage are not able to feel the pain, and that leads to the wounds, which was earlier associated with the stigma, because people would lose their digits during infection, and so
dermis, it mostly stays in the dermis, epidermis.
It goes to the peripheral nerves. It damages the nerves and causes demyelination. Demyelination.
So is there a correlation between a diabetic caused neuropathy and...
It's a very good actually model actually for in the armadillo, the nerves that I was showing, that is used for diabetic neuropathy also.
So there is a correlation there,
yes,
clearly, because you are damaging the nerve tissue,
yes, sir.
And then infecting it on the top of it.
Yes, so it is a model for the peripheral neuropathy research also.
Okay.
No, they develop disease. They even develop the loss of sensation. They are the only model of leprosy with the clinical presentation also.
Okay, if there are no more questions, I would like to thank Dr. Pushpendra Singh again.
Thank you very much, and thanks to the organizers.
We'll move on to the next lecture by Jeevan Jyoti, Dr. Jeevan Jyoti Panda. It will be online.
Yes, hello. Yeah, can you see me?
Yeah, we can see your slides.
Should I start?
Uh, can you listen ours?
Yeah, I can just listen to you guys. So can I start?
Yeah, sure.
Yeah. So, good morning, everyone. First of all, I would like to thank Dr. Ambati, Dr. Anil, and Dr. Sugnakar for the kind invite and for giving me this opportunity to share my work here. So, today I'll be talking about some of the brain delivery or neuro nanotherapeutic space research work which we carry out here at INST, the Institute of Nanoscience and Technology, Mohali, a autonomous institute under DST, which is dedicated completely towards the research in nanoscience and technology. So you all would agree with me that brain is the master organ. It is the most important organ and it controls anything and everything we do. So it really gets very important that it should be guarded and protected well. And this job is done by the blood-brain barrier. This barrier is formed by the tight layer of endothelial cells which lines the inner surface of brain capillaries. And it's a semi-permeable barrier, which means it allows some molecules to pass through but restricts the entry of others. And if you ask me, the brain cells or brain tissues really love it because it protects them from harmful and noxious substances. But when we talk about brain drug delivery or neurotherapeutics, it creates a major bottleneck. Why so? Because it has been shown that 98% of small molecular drugs and almost 100% of large molecular drugs cannot cross through the barrier. So it really gives a nightmare to the pharmaceutical scientists working in the field of brain therapy. Though different strategies are being used for ferrying molecules through the barrier, which involves the transnasal drug delivery, in which the drug molecules are instilled in the nasal cavity from where they can directly reach the brain tissues. Or some people tried transcranial or invasive strategies in which the drug is directly injected into the brain tissues, or sometimes we may go for non-specific disruption of the blood-brain barrier, which involves breaking the barriers using molecules like osmolytes, or in many cases, the drug molecules are themselves modified with chemically modified. So though they have given us some success, but they come up with their own demerits. Like if you talk about transnasal drug delivery, if a person is having cold or allergy, this route cannot be used. And of course, if someone is unconscious, we cannot use this route. And when we talk about invasive strategies, they are kind of not very good because they lack patient compliance. We need the involvement of experts, doctors are involved, and many times they come up with lots of side effects. And when we non-specifically disrupt the blood-brain barrier, you can imagine that it may also allow other harmful substances to gain entry into the neuronal tissues, which can be really dangerous for the neurons. And if we chemically modify, we may compromise with the activity of the molecule. Thus, it really becomes very important that we should develop different strategies through which we can allow molecules to cross through this blood-brain barrier to reach the brain tissues in an effective concentration so that different disorders can be treated well. So as nanotechnology has shown different, it has been really promising in many aspects of drug delivery, especially in case of cancer. There are many marketed formulations. So we are really hopeful that this can also be helpful in the field of brain therapy, brain drug delivery, and for ferrying molecules across the blood-brain barrier. So my group basically works in the field of peptide nanostructures. We work in the field of peptide, amino acid, and protein-based nanostructures. And why so? Because we don't generally, this is the base because peptides are highly biocompatible. They are our own molecules, so you can completely rely on them. And a lot of literature shows that they can themselves form these kind of nanostructures. They can self-assemble to form variety of nanostructures. And since we have lots of 20 amino acids naturally occurring and many non-naturally occurring amino acids, when we use peptides as a template to synthesize the nanostructures, we can actually expect the development of the synthesis of very tunable nanostructures and which have lots of functional groups which can be used for targeting different receptors present in the disease tissues. And the most important motivation here when we talk about blood-brain barrier delivery using peptides is that there are a variety of peptides and amino acids which can inherently cross through the barrier. So you don't need to, tag them with something. And if we choose and make our nanostructures using those kind of peptides, amino acid or proteins, our nanostructures can inherently or by themselves can cross through the barrier. So this will make our job very easy. So having said that, this is our kind of a strategy which we are following. We have amino acids and we synthesize them into different peptides in our lab, in our institute, and then we allow these we incorporate design elements in them so that they can self-assemble to form variety of nanostructures. And then we characterize these nanostructures for their brain delivery potential using different models, the in vitro parallel artificial membrane permeability assay, in which we can test the BBB permeability of molecules. Then we have cellular models, cell-based co-culture models, and then we use in vivo imaging system to test the brain delivery of potency of these molecules. And finally, we try to use them in different brain disorders like glioma, Alzheimer's, stroke, etc. So this slide kind of summarizes our lab's work, what we are doing currently. And today I will be showing you some of the examples. Basically, I'll be talking about a few examples in which we have used these structures for glioma therapy and in neurodegenerative disorders, because we may not have time to discuss the other works. So just to tell you, like for the students, glioma is a tumor that affects the brain and spinal cord, and it suffers from very poor patient survival rate. And if we talk about glioblastoma, that has very less percentage of 5-year survival. And this disease also suffers from BBB restrictions. So in our lab, we are kind of trying to develop theranostic systems for glioma. Theranostics means in which we are trying to, in some examples I'll show you, where we are trying to develop both nanostructures which carries both therapeutic and imaging or diagnostic modalities in them. So they serve everything in a single platter. So we call them theranostic systems. So for glioma therapy, we tried target-specific and stimuli-responsive nanostructures. We developed the pH-responsive vesicles. Then we also developed pH-responsive nanobowls. I will be discussing about this work. We also tried to develop light-responsive structures, wherein we can use different types of light as lights of different wavelengths as a stimuli to release the molecules in the glioma tissues. Then we have redox responsive structures, and in a very interesting work, we have multi-stimuli responsive nanostructures. These nanostructures actually responded to many different types of stimulus, and I will discuss about this work as well. So this is about the nano bowls which we made. You can see these are very beautiful nano bowl-like structure, and here we use microfluidic based self-assembly to construct these bowls. And you can see these are very, nicely formed structures in which we can load drug molecules. And these bowls were being formed by a modified amino acid, the FMOX trityl OH. And since the bowls, we wanted to cap it to further protect the drug. So we used another amino acid as a capping agent. So these are our bowls after being capped with the second amino acid. And interestingly, our amino acids carry pH responsive moieties, the trityl and BOC group. So when we subjected these bowls to different pH conditions, pH perturbations, we found that they form this kind of pores through which the drug molecules can be released. So these are our bowls after being loaded with doxorubicin, the anti-cancer drug. And then we also tested for their efficacy in glioma cells where they demonstrated enhanced efficacy with a reduction in IC50 values. And we further tested their efficacy in tumor models in which glioma tumor models, and you can see that when we have the bowels, there is a significant reduction in the tumor size, which shows that this pH responsive bowels can act as very good delivery vehicles for the glioma drugs. We also use these bowls for dual drug delivery for combined chemo and phototherapeutic activities, photodynamic activities. So for that, we retain doxorubicin. But for having photodynamic activity, we included curcumin into the bowls. So curcumin has got anti-cancer activity. Along with that, it has got photodynamic activity. It responds to blue light. So, and we tested for the efficacy of this cellular uptake and efficacy of these structures in glioma models, both 2D and 3D cellular models. You can see this is the 3D model in which the cells were seen to take up the bowels and they're kind of showing the fluorescence. And we found that when we expose the cells to the light, they exhibited enhanced efficacy in the glioma cells. So these systems acted as a dual combined chemo and photodynamic therapy for the glioma cells. These are a few examples in which we worked on pH responsive dipeptide nanostructures. Here we used dipeptide, cationic dipeptide nanostructures, and here we used cysteine phenylalanine based nanostructures for redox responsive drug delivery. And anyone interested can go through it. And due to in the interest of time, I'm kind of not going to share the details of this work. Yeah, so this is another work in which I was talking about multi-stimuli responsive nanostructures. This is a very interesting piece of work in which we found that a single peptide monomer, this one is being designed by us. So this peptide monomer, we found that when we tweak the assembling condition, this peptide monomer can form 3 different types of structures. And it can shuttle between these 3 different types of structures. So where these are like 2D structures, it can shuttle between 2D and 3-dimensional structures. So this is really interesting. It formed sheets, fibers, and spherical structure, vesicular structures. And to our interest, these structures also exhibited inherent fluorescence. So this is very interesting as because we have a structure which can shuttle between 3 different shapes, morphologies, and has got inherent fluorescence. And anyone working in the field of drug delivery will agree that shape is a very important parameter because it determines the ultimate fate of the structures inside the cells and inside the tissues. So we decided to determine their cellular uptake behavior. And we did this in presence of the endocytotic and phagocytotic inhibitors. And we really found that to our expectations, the structures exhibited, shape-dependent cellular uptake behavior. You can see where the fibers and the sheets exhibited phagocytotic or macropinocytotic based uptake inside the cells. The vesicles exhibited clathrin-mediated endocytotic uptake behavior. So having done that, we just wanted to see if that affects their efficacy, anti-cancer efficacy. Those studies were done in glioma cells. So to our expectations, we found that the nanosheets were most effective. The drug-loaded nanosheets were most effective in killing the cells as compared to the other 2 structures. This may be corroborated to their higher uptake and they exhibited enhanced cellular uptake and they exhibited the maximum cell killing efficacy. So this shows that how we can tune the drug delivery potency of structures using our peptide-based nanostructure based on their shapes. This is another interesting piece of work where we tried to develop theranostic nanostructures. And here we use levodopa because this is an anti-Parkinsonian drug and it can cross through the blood-brain barrier itself. So we found that it can self-polymerize into fluorescent polylevodopamine nanostructures. And these structures, literature shows that polydopamine nanostructures, they exhibit photodynamic activities. In order to further enhance their activity and in order to further make them multifunctional theranostic system for glioma, we conjugated them with an peptide, this peptide, I do not have the sequence here. So this is being synthesized, designed in our lab. It carries both glioma targeting and anti-glioma activity. So it's a chimeric peptide. And we attach this to the nanostructures. Along with that, we loaded it with the anti-cancer drug doxorubicin and another NIR responsive dye, indocyanin green. So this is a complete multifunctional nanostructure which can cross through the blood-brain barrier, can target glioma cells, tissues, and can exhibit combined photo and chemotherapy. So these are our structures, and I just want to emphasize here, these are our structures and they were self-fluorescent in nature. So you can see these are the 3D spheroids which are glowing bright after taking up the cells, so they can act as imaging and therapeutic modalities. So we tested that brain delivery here using IVIS. You can see they are targeting the brain and can stay there for some time to act as a brain delivery agent. And then, yeah, so these structures also exhibited combined chemo and phototherapy, and they exhibited their photodynamic and photothermal activity in response to NIR laser, the 808 laser. You can see these are the structures where you see in the final formulation, we obtain the maximum cell killing, which shows that they're really effective in killing the cells. Both 2D and 3D cellular models we tested, and we found that they're efficacious. Now, similarly, we also developed levodopa. We used as a template to synthesize gold nanoflowers. This you can see very beautiful gold nanoflowers being synthesized on levodopa nanotubes. And these flowers exhibited Raman signaling based imaging modalities in glioma cells. And we also use them like in our previous example, we use them for combined chemo and photodynamic therapy of gliomas. So those are some of the examples where we used our structures for glioma therapy. And now I will show you one or two examples where we tried to use our structures for anti-amyloid therapy. Here we developed different types of nanostructures which can cross through the blood-brain barrier to exhibit anti-amyloid effect. So amyloid or Alzheimer's is a neurodegenerative disorder. It's a leading cause of dementia. It's a progressive neurodegenerative disorder, and it majorly occurs due to the accumulation of amyloid, that amyloid APP, the amyloid precursor peptide, it wrongly, when it non-conventionally get cleaved, it forms A beta 40 and A beta 42, and these peptides, polypeptides, accumulate, they form aggregated plaques inside the brain tissues, and these kind of triggers lots of signaling cascades, which finally forms a tau fibrillation and finally leads to the death of the neurons. This shows that any molecule if any molecule can prevent the aggregation of this amyloid polypeptides, they can act as potential a neurotherapeutic agent for the disorder. And if these molecules can cross through the blood-brain barrier, that would be really a boon for the system. So keeping that in mind, we try to develop theranostic nanostructures for amyloids, Alzheimer's, and here we use tryptophan. So tryptophan we chose because it can cross through the blood-brain barrier. It can use LAT transporters to gain entry into the brain tissues. And various literature shows that tryptophan intake improves memory and it reduces amyloid plaques, and it has got antidepressant effect. So, and several tryptophan conjugates have been shown to carry anti-amyloid effects. How many what time I have? How much minutes? Sorry?
Yeah, 2 more minutes.
2 minutes, okay. So I have to rest. So we use tryptophan and we try to synthesize tryptophan nanostructures using hydrothermal based processing. And you can see these are our nanostructures, and like our previous case, they were self-fluorescent in nature. And we tested their anti-amyloid effect. First, we developed different in vitro models, in vitro peptide-based models in our lab. So this is FIFI, which is derived from A beta 42 peptide, and this form fibers and can be used as an amyloid model. And this is A beta 42 aggregates. So you can see that our structures could disrupt these aggregates, and you can see here that after 42 hours the aggregates are completely gone. And then we tested for their cellular uptake behavior. Since they're cell fluorescent, they were taken up by the cells, and they could label the brain tissues and the cells, because we found that they can interact with the fibrils. Here you can see that these structures interacted with the fibers, FIFI fibers. So somehow they were able to tag the fibers, and since they're fluorescent, we can tag them inside the plaques and can be used for imaging these plaques. So we next tested any anti-amyloid or anti-AD drugs should be able to protect the neuronal tissues from the toxic effects of the amyloid fibers. So we tested that. We used neuronal cells for that. And we found that our structures can really protect. You can see this is a live dead assay where we can see that maximum cells are viable when we tested. We use our structures with the fibers. So they can protect, and they help the neuronal cells to revive they revived it. Actually, they helped them to gain their viability. So they could protect them from amyloid fiber induced toxicity. And we also tested for their efficacy in brain animal amyloid animal model. So for that, we injected streptozotocin into the animals, and we tested for their behavior deficit using Morris water maze, where you train the animals to reach to find some hidden platform. And if they have amyloids, they cannot do that after many training if they have AD. But if they are normal, they can find it after certain training. So we found that our structures, they're almost like control, where this is the streptozotocin, the animal carrying AD, and this is after treatment with our structures. So they help the animals to regain their cognitive and cognitive behavior, and also they reduce the amyloid burden in the brain tissues. They can tag the amyloid plaques in the brain tissues in the in vivo model, and they also help reduce the burden of the amyloid plaques in the brain tissues. So they acted as a theranostic system for the disease. As I mentioned, that this conjugates dopamine tryptophan conjugates can also act as anti-amyloid agents. So we developed a conjugate of tryptophan with another anti-amyloid molecule, dopamine. So these two also we synthesized using hydrothermal processing. And like in our previous case, you can see that the structures are kind of, the fibers are kind of very nicely disaggregated in presence of our hybrid nanostructures. And like other case, you can see that they are kind of tagging the fibers. These are the amyloid fibers, and this is the ACM image showing that how beautifully they are sitting onto the fibers, and they can be taken up by the cells. They help the cells to glow. This is without any dye. You can see the neural cells are glowing. And we also tested for their efficacy in animal and cellular models, where we found that they could protect the cells from the fiber induced toxicity, and also help the animal in regaining their cognitive behavior. Similarly, we also used another very interesting system in which we developed selenium nanostructures. Selenium has got, yes, yeah, I'm done almost, 1 or 2 page slides. So here we used a hybrid structure using selenium and a dipeptide nanoparticle, and selenium has got anti-amyloid effects. So we expected that these structures would carry anti-amyloid propensity. So we tested them in 3 different amyloid models. Both the FIFI and this A beta was still there. And then we also used another hexapeptide, which is related to tau fibrillization. So these structures can inhibit both A beta and tau fibrillization. So this is a multifarious system. And we also tested for their efficacy in streptozotocin induced cellular models, where they have been found to protect the neuronal cells from A beta fiber induced toxicity, both A beta and tau fibril induced toxicity. So, which means that these structures are multifarious in nature. They can stop both A beta and tau fibrillization. So, if you study amyloid literature, both the proteins are involved in forming A beta forms amyloid plaques and tau forms neurofibrillary triangles. Both lead to the death of the neurons. And this we tested their blood-brain barrier delivery and brain delivery potential. And like other case, we also tested for their efficacy in amyloid animal and cellular models. So thus to conclude, the BBB, the blood-brain barrier, creates a huge bottleneck in the delivery of neuronal therapeutics, and our peptide-based nanostructures can really breach the blood-brain barrier to reach the brain tissues to deliver potent neurotherapeutics, and they can be used as a theranostic system to treat these different neuronal disorders. So with this, I would like to thank my students. These are the students, Taru Sonika, whose work I presented here, and all my collaborators, Professor Kundu, NCCS Pune, where I carried out in vivo tumor studies, Professor Chauhan, and Dr. Subhash Shukla for animal studies with AD, and Professor Uday Kamtela of University of Colorado, where I carried out some tumor-related and the phototheranostic studies, and my in-house collaborators, Dr. Bhanu and Dr. Manis. And also thanks are due to the funding agencies for all the support. And thank you everyone for your kind attention.
Thank you very much, Dr. Panda, for a very interesting topic.
Yeah, thank
Since we are running a little bit late, maybe we can take one question if there is one. Any from the students? Yeah.
I'm curious about this blood-brain barrier. While even the simple molecule like magnesium cannot go through well, you have to chelate that with threonine to make it more permeable, while the red blood cells can go through. So what is the mechanism there that allows cellular structure to pass through, but even the small molecule like magnesium is not easy to go through?
Yes, it's very interesting question. Actually, for when we talk about cellular structures, they gain entry into the BBB through transcytosis. So that is also a mechanism, little bit, microparticles can also adopt to cross through the blood-brain barrier. So this is specific, but for WBCs and for, as you mentioned, for the cells, they allow this transcytosis mechanism to cross through the blood-brain barrier. But small molecules, there are efflux transporters which throws them out so they cannot cross through. So yeah, I hope it answers your question.
Yeah, one more question from.
Hi, ma'am. Actually, I'm working in the area of glioma only. So I was just, curious. Usually, when we check for animal models or cell lines, in glioma, the major standard drug that we use is temozolomide, and it's an FDA approval drug, which I think in the year of 2005 it got its approval. But
Right.
whenever we go for cell lines or in vivo model studies, we develop the temozolomide resistant models. So, ma'am, I just wanted to know why did you choose doxorubicin in that case?
Yeah, actually, temozolomide, sometimes when we use nanostructures, we found we used temozolomide, but we had some stability issues with the drug. So, and doxorubicin is a well-known anti-cancer drug, so we thought of using it, and it has got some fluorescence, that will also help us in tracking the systems inside the brain, brain cells or brain tissues. But yeah, of course, we are we'll be using temozolamides in some other models.
Yeah, maybe because temozolomide is a lipophilic molecule, so if you can try with that, I think it can be also a nice data for
Yeah,
that in that case,
it is
ma'am.
approved. We have actually, I didn't present that where we have some systems where we have used temozolomide. So that work, we have plans for that.
Thank you, ma'am. Thank you so much. It was
Maybe I
appreciable.
think you should communicate with her on email and get all your doubts.
Sure,
Thank you
yeah.
very much, Dr. Panda. Very nice
Thank you.
talk. And
Yeah,
we will
thank
move on to our next lecture. It It is by Professor Bipin Nair from Amrita Vishwavidyapith, Kollam, Kerala. And he will be talking on novel approaches to target multi-drug resistant Pseudomonas aeruginosa. Dr. Bipin.
Slide change. Sorry. Okay. Still morning. Good morning. And it's a real pleasure to be here. And I wanted to thank BioClues, Prashant, and Dr. Kavi Kishore and the organizers for giving me this opportunity to present some of the work that we've been doing at Amrita School of Biotechnology, trying to look at bacteriophages as a tool to target AMR and in escape pathogens. So a lot of acronyms in there, and I'll come to that. But this meeting is a good midpoint because we just finished observing World Bacteriophage Day or Week, October 22nd to 28th. And then coming up November 18th to 24th, we have World Antimicrobial Awareness Week. That's what AMR stands for. And then escape pathogens. So we'll talk about a little bit of all of that. Yep, thank you. So this audience doesn't need much of an introduction to this concern that is now there, that there is a global increase in multi-drug resistant infections. In fact, Dr. D.N. Rao also alluded to that yesterday in his talk. And the problem is more complex because there is very sluggish development of antimicrobials. The pharma biotech sector is not interested in developing new drugs against drug-resistant microbes. And then all other approaches, antibiotic stewardship and the generation of other effective therapies, including vaccines or gene editing or phage therapy, all of that has had very limited outcomes or limited success. So what exactly is this global antimicrobial resistance crisis? Looking at it right now, AMR or antimicrobial resistance is attributed to about 700,000 deaths, and that is also an underestimation. In fact, the World Health Organization has projected that if we are not careful about this, by the year 2050, about 25 years from now, that number can go to 10 million deaths a year, way above what happens with cancer or diabetes or any other form of disease. And of that 10 million, the contribution from India may be 2 million deaths per year. So India does need to take care of this crisis as it is coming up. It is here already. It is almost called a silent pandemic coming on the heels of the pandemic that we just had. Antimicrobial resistance is a big, big concern. And what is central to that are these escape pathogens, as they are called: Escherichia coli, Staphylococcus, Klebsiella, Acinetobacter, Pseudomonas, and Enterococcus. So the problem is there is increasing drug resistance, not many novel antibiotics being discovered. So we are looking at what can be called the end of the antibiotic era. And unless we do something about it, that may be a major problem. So of these escape pathogens, we decided to focus on Pseudomonas aeruginosa, because the World Health Organization a few years ago classified Pseudomonas as a critical priority antibiotic resistant pathogen. And the way Pseudomonas is able to do this, it has intrinsic resistance to most of the antibiotics that are available, and it is also rapidly developing resistance to the last line of antibiotics, including carbapenems and polymyxins. How Pseudomonas does that is by very effective use of these efflux pumps. So whatever antibiotics you administer, it is very effectively able to pump it out with these efflux pumps. And the resistome, the collection of genes that are responsible for resistance in Pseudomonas, that has very high plasticity, making the problem even more complex. And people have been looking at different approaches to counter that, and bacteriophages specific for Pseudomonas has been a line of attack, and most of these Pseudomonas phages that have been characterized belong to the Cordovirales family of double-stranded DNA phages. A lot of work being done across the globe, not much in India, but at least in Europe, in France, in South Korea, in Russia, in Belgium, for example, a considerable amount of work looking at bacteriophages specific for Pseudomonas and how these lytic phages could probably be applied to counter this problem. For our part, we tried to, or we came into the picture when we were funded by the Bill and Melinda Gates Foundation in collaboration with DBT Biotek to look at the problem of sanitation and wastewater, where infection with bacteria or parasites and also malodor causing bacteria are a major concern. And we thought of utilizing bacteriophages along with a microbial consortium or enzymes or bacteriocins, small molecules from natural sources, and producing what we call a lytics broadcasting system that can be used to effectively administer and counter pathogenic bacteria. And so we set up this protocol to isolate bacteriophages that could be specific for the gram-negative pathogens that we are looking at. Sewage is a rich source of these phages, enriching these phages with target bacteria and going through a process where we can ultimately characterize these phages, sequence them, annotate them, and have an idea of the specific nature of these phages and the hosts that they can attack. We now have a reasonably good library of bacteriophages specific for a lot of clinical and environmental pathogens: E. coli, Salmonella, Shigella, Vibrio, Klebsiella, Pseudomonas, and the likes. And what we've been able to do is go through characterization, identify whether they are lytic or lysogenic in nature, look at their tropism, narrow or broad spectrum, look at their melting curve, look at the genome, identify them as DNA or RNA phages, and then go through, in this case, specifically identify phages that can infect multi-drug resistant Pseudomonas strains that we have isolated, and we now have a good collection of multi-drug resistant Pseudomonas strains that we have obtained from different hospitals and clinics across the country. And here is what we are concerned about. These are strains, multi-drug resistant. If you look at that picture, R standing for resistant, all these strains isolated from different sources are resistant to most of the antibiotics that you can look at, except for polymyxin where they are, or for colistin where they are sensitive. Otherwise, you see resistance all across. So that is a major concern, and what can we do about it? So what we've done is gone through and work done by a graduate student in the lab, Natasha Menon, was able to bring about the nice isolation and characterization of a phage which we called AMP2. You can see the plaques formed by the AMP2 phage on a lawn of Pseudomonas, a clear center with a turbid edge. That morphology, and then we, in collaboration with InSTEM in Bangalore, we were able to get a nice picture of that phage, the head and the short tail, and a conveyor belt-like organization of phages on a bacterial cell attacking the phage, and then carrying out the characterization, the phenotypic characterization, seeing a latent phase for about 20 minutes, followed by a burst. And when we looked at the killing of Pseudomonas, the wild-type Pseudomonas PA01, at a multiplicity of infection 0.1, 1, and 10, very nicely able to demonstrate killing of the bacterium, lysis of the bacterium, with multiplicity of infection 0.1, 1, or 10. Went through and did further molecular characterization, identified a 73 kb genome, and then also were able to identify that there was considerable similarity with Luseptima viridae, two phages, and then but distinct enough for us to characterize it as a novel bacteriophage that we had isolated. It was also interesting that in the collection of multi-drug resistant Pseudomonas that we had, AMP2 was able to bring about lysis, able to bring about lysis of about 60% of those MDR strains, and this gives you a picture of what is the effect or what is the activity of AMP2 against all these multi-drug resistant or sensitive Pseudomonas. So the question that came up, and what we were looking at is, since you're looking at antibiotic resistance, would these phages that we have isolated, would the strains become resistant to these phages as well? And in collaboration with Victor Nizet's lab at the University of California, San Diego, again, Natasha Menon carried out some elegant studies using these bacteriophages AMP2 and then MAT and CAT. And what we found is that there were PA01 strains that showed resistance to some of these. And what was characteristic of these phages is that in vitro treatment of PA01 with these phages resulted in phage-resistant mutants that produced this pigment pyomelanin, and you can clearly see that increased production of pyomelanin in these strains that were resistant to the phages. And what was also very interesting for us is that these pyomelaninic mutants, which are phage resistant, also harbor a large genomic deletion that renders them less virulent. So they are resistant to the phage, but they are also less virulent than what we would have expected. These studies have been published in EMsphere and Antimicrobial Agents and Chemotherapy, giving us a good foothold on what could be done with this. We also started looking at another problematic escape pathogen, which is Klebsiella pneumoniae. It's a gram-negative, facultative anaerobe that is an opportunistic pathogen responsible for a lot of nosocomial infections across the country. In fact, clinicians tell us that between Pseudomonas and Klebsiella, these are the major problem causing multi-drug resistant infections that they are worried about. And a lot of these are present in the country, and what we have also done is try to characterize the same in collaboration with the Rajiv Gandhi Center for Biotechnology in Trivandrum. We were also able to obtain a TEM image of the KP3 phage, a Klebsiella specific phage, and also picture of these phages attacking a bacterium. One other escape pathogen that we have begun to look at is Acinetobacter baumannii, commonly found in healthcare facilities. Again, also a major concern for clinics and hospitals across the country because it is an opportunistic pathogen. And what we have been trying to do also is that because detecting multi-drug resistant infections is a problem, we are in the process of creating a phage array kind of detection system where we use the phages. Since we have characterized them, we are able to very specifically identify in the span of about 4 to 6 hours, instead of taking the 24 to 48 hours for culturing the bacterial strains and identifying them. Within 4 to 6 hours, we would be able to bring about identification of a specific multi-drug resistant strain using the phages that we have. I won't go into the details in the interest of time, but that brings us to the point that I wanted to share with you. Can we look at phages as a good alternative or as in a supporting role to antibiotics so that we can more effectively counter the problem of multi-drug resistant infections? So when you look at phages as such, they are highly specific. That's the biggest advantage. They will only infect the bacterial host for which they have been identified. There is no cross-reactivity, not with other species, not with other bacterial species even. And instead of that, in case of antibiotics, of course, there is low specificity, there is broad spectrum activity. So that is a problem that phages would be useful to encounter or to tackle that problem. If you look at the pharmacokinetics, these phages will replicate only at the site of infection, not anywhere else. However, in case of antibiotics, we know they are metabolized and they are eliminated over time. To the best of our knowledge, there are no serious adverse effects in the use of phages. However, again, as we know, antibiotics are riddled with all these problems, intestinal disorders, secondary infections. With respect to resistance, that resistance to the specific, if you are using that to treat an infection, resistance can only arise in target bacteria. However, we know that non-target bacteria can become resistant in the use of antibiotics. And what is a bigger concern that we talked about earlier, the development of new antibiotics may be slow to come by. You can very rapidly identify, isolate, identify, and characterize, and be able to use these phages for specific bacterial infections. So there are some problems, but if you're looking at phage therapy, yes, there is a narrow host range. What we can do is, if there are multiple infections, and I'll give you some examples later on, if there are multiple infections, you can use a phage cocktail. Multiple phages specific for the different organisms that may be causing a disease. Then there is the problem that you may not have been able to purify those phage preparations effectively. You can use a lot of technology that is now available to get highly purified preparations of these phages. That accounts for the poor stability and viability also. And then it's essential to understand whether these are lytic or lysogenic phages, and especially for phage therapy, we would have to focus on lytic phages and not on the lysogenic type. And there have been exaggerated claims of how effective phage therapy has been, but that has been effectively practiced, especially in Eastern Europe, over 100 years where phage therapy has been in use. We here need to have more research so that we can classify, we can characterize, and we can look at the efficacy of these phages against specific bacterial pathogens, look at the possible side effects and so on. And then there have not been enough clinical trials. So to establish this scientific proof of the efficacy, we need double-blinded, placebo, carefully controlled clinical trials so that we can establish that that kind of phage therapy that we are thinking about is possible, and that is what we are looking at now. We have tied up with a company, Vitalis Phage Therapy, that's a company established in Delhi, and they can claim to have brought phage therapy to India. The founder of Vitalis, Pranav Johri, was the first Indian patient to have received phage therapy after 5 failed antibiotic treatments, where he traveled to Sibelius in Georgia, and at the Eliava Institute there was able to obtain phage therapy for his antibiotic resistant infection. And he came back to India and said, this needs to happen here. He has set up Vitalis phage therapy, and they are now very effectively bringing that technology to India directly from Eliava and bringing about the possible treatment of patients infected with multi-drug resistant infections on a personalized level, on a compassionate level basis, but without having to travel internationally and go elsewhere for that treatment. They have also initiated sensitivity testing of these phages and are now in the process of collaborating with others, and for example, looking at the possibility of having referral centers like our hospitals in Kochi and Delhi to bring about a possible phage therapy initiative here in India. And I've been told that there are at least 150 such cases where phage therapy has been effectively used, and that is something positive that we can look forward to. So the take-home message that I would like to leave you with is that there is a sustained effort to that needs to supplement this repertoire of specific as well as broad-spectrum bacteriophages, which can then be considered as an effective and essential tool to be able to counter that unmet need in an effective and efficient manner and confront this global AMR crisis that we have on hand. Finally, this is where we are located, the beautiful backwaters of Kerala. If you have not had the opportunity, you are welcome to come here. This is what I like to leave as a teaser slide, because for those of you who have to travel a long distance on the roads, fighting traffic, all I have to do is to cross that bridge from home to the labs where we are. So please come. Thank you.
Thank you very much, Dr. Bipin, for sticking to the time. And we can take 2 questions. We are running late.
Sorry.
It's very interesting talk.
Thank you.
Okay, I appreciate. But I have 2 simple comments. The first one is majority of these bacterial species, they have an adaptive immune system, like the CRISPR-Cas clusters, over a period of time, sure, they will develop resistance against these viruses. So in long run, this may not work. I mean, this is my comment. Just you can comment on this point. This is one. The second point is that you mentioned somewhere that they are very specific. This
yes.
Virus to bacteriophage to bacteria. So can you comment on this point, how they became over a period of, specific to specific bacterial species? So these are the 2 points, and you may please, I'm excited to.
I'll take the second question first.
Okay.
From time immemorial, as from the earliest times when phages were first discovered, 1917, the host bacterium for the phage that we are looking at has always been very specific. There has never been any cross-reactivity. In fact, a Pseudomonas phage going to a Staphylococcus aureus is never known. Within the gram-negative, gram-positive species also, there has always been that specificity.
So these determinants are already characterized,
Definitely,
very well known?
definitely. As far as developing phage resistance, we started to look at that, trying to characterize, do phages become resistant? I mean, do bacteria become resistant to phages? And there's no telling, there hasn't been enough characterization of that. The positive point that we can take away from these studies that we observed is that indeed the Pseudomonas strains that we were looking at did develop resistance to our phages, but developed that potential to have this biomelanin production. And what we found is that they were more sensitive. So the bacterium was losing some of its capability to be virulent in developing phage resistance. We don't know what the correlation is, but at least that is something that we can look forward to. But the other aspect of what we can look forward to is that there have been hundreds of cases across the globe where doctors have given up, including Jory, where doctors said there's nothing that we can do here in India and anywhere in the world, but you can just manage your symptoms. Till he personally did research, went to Eastern Europe, and then got treated for these. He had 4 different strains of multi-drug resistant organisms, and they were able to apply a cocktail of phages and treat that within 3 months of this treatment. He was completely free.
The point is just this in your screening, have you found any cyanophages, bacteriophages which are specific to cyanobacteria?
We haven't been looking at that. We have focused only on these escape pathogens.
Thank you very much. Thank you.
Thanks. Sir.
It is a wonderful talk on bacteriophages. Is there anyone among your team working on M13 phase, sir?
No, we have not.
No, we are interested to cultivate in Vignan University.
Okay.
Yeah. So we are having a course on phase display. So wherein we want to show to our students this M13 phase.
Yeah, we have not looked at it. We isolate our own phages specific for any of these escape pathogens and then go about characterizing those and looking at their activity.
Sir, sir, I thoroughly enjoyed your presentation. It was like a scientific story, but I missed out, I think, at one place, and one is my question. That is, so the question, 2 questions I have. Second, I will ask first because I think it will easy to answer, and the first one can be a method, so we can talk in personal. So my question is, you have already talked about the phage therapy. So as far as I know, the antibiotic, it is a drug in the form. It is in the drug form. It is given to the person. But how phage therapy can be given? It is in the drug form, or it is some liquid form, or what?
Yeah, so as of now, what has been done, and Pranav Johri and his team at Vitalis have made that kind of an arrangement in collaboration with Ilyava Institute in Georgia, where it can be imported into India as a drug preparation. You go through, get CDSCO clearance for bringing in this formulation as a drug. It may be a single phage, it may be a cocktail. Once that infection is identified, they will find out what is the nature of the infection, what phages are necessary, how it should be administered, what it should be.
Because the antimicrobial drugs are certainly chemical molecules, and these are the kind of living organisms. So I was just curious, like how to treat them with the living organisms.
So as of now, it is mostly topical. In case of wound infections, or it can be as suppositories, or it can be applied in a way that You are not actually injecting it. But there have been cases, especially, for example, one very well-known established case in California, where they had to actually administer a cocktail of phages and intravenously. So it's possible, but there may be complications. We'll have to deal with it as it comes up.
So
Sir,
second question I will ask you in personal.
Okay.
Thank
Thank you.
Sir, assuming that, excuse me, sir.
Sir, sorry.
Assuming that it has been given systemically,
Mm.
phages, would there be any effect of our natural system to tackle that, or to prevent its action?
The
Like antibodies.
Yeah, the concern has been that you are administering phages, they would attack and bring about lysis of bacterial cells. You may have lipopolysaccharide, for example. But that has never been a concern, at least in the cases that have been administered phage therapy. That has not become a major complication as far as an immune response is concerned. Sure. Yeah.
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 Thank you. It seems there are a lot more questions, and in the paucity of time, I think.
Thank you. Thank you.
We must
Thank
thank Dr. Vipin. Thank you very much, sir. Now I'll hand over the mic to the organizers.
Thank you, Krishna Mohan sir. Thank you for very effectively organizing the fourth technical session. We once again thank all the three speakers for this session, and I request Chairperson Dr. Krishna Mohan sir to felicitate our speakers, Dr. Pushpendra Singh sir from ICMR, National Institute of Research in Tribal Health. Sir, thank you for your wonderful lecture, and please accept our felicitation on behalf of the organizing committee. Thank you, sir. I now request Vipinji Nair, sir, also to please accept our felicitation. Thank you. We will now move to the next segment. Before lunch, we have this oral presentations 11 to 20, and this oral presentation session will be chaired by Professor D.T. Nikam sir, and we have Dr. Hamza, Associate Professor from Malaysia University. May I have the chairpersons, please? And the rapporteur for the session will be Dr. Sandeep Chauhan. Sir, Sandeep sir, I request you to please take over.
Thank you, madam. Thank you,
So
madam.
we have Dr. Zulhazman Hamza. Yeah.
So, I request the following persons to meet me as soon as possible. Simi, PG, MIS, Rotak. JJ Venkat Nagmani, madam, IICT. Pavan, Niti, deemed to be university. Vivek Kumar Sharma from AIMS Raj. Please. PVV Sivakrishna from Vignan deemed to be university, and Pri Srinivasa Babu, Vignan deemed to be university. Please come and meet me as soon as possible. with respect to oral presentation. ఒకటి ఇచ్చారు దట్ ఈస్ ఓకే అది ఎవరికి ఇవ్వాలి So I would request Noor Fazlani Shapova to come here for a seminar for oral presentation, and the topic is constant flow rate feeding fed batch fermentation kinetic modeling of DHA production by Schizochytrium species. Yes.
Slide changer.
Yeah. Can you
నిన్నటి వాళ్ళు నేను ఇది మార్చాను ఆన్లైన్ అని మార్చి కట్ చేసి సీరియల్ నెంబర్ అని రాశాను మీరు తర్వాత ఆయనకి పేర్లు ఇవ్వండి జస్ట్ గివ్ హిమ్ విచ్ నేమ్ సో దట్ హి విల్ నోట్ డౌన్ అండ్ మార్క్
Okay, assalamu alaikum and very good afternoon everyone. I think afternoon. Okay, my name is Dr. Noor Fazliani from University Malaysia Kelantan. Before I start, let me show you a short video related to my university. That is about my university. Okay, I'm also one of a director of GRID. This is a Gold Rate Earth Material and Technology Center. Okay, let me. Okay, my presentation is related, maybe different from the topic today. It's about fat to model related with the fat bed fermentation using constant flow rate feeding in fat batch fermentation process. Then after I formulate the model, I identify the kinetic parameters and validate with the experimental study. Okay, this is the research background. As you know, the demand of DHA grow global due to the potential to prevent the chronic disease. Okay, Schizotrium species is one the species that can produce a high amount of DHA, but we need to do the fermentation process to extract the DHA, and one of the best way to get the DHA production is from the fat batch fermentation process. Okay, why fat batch fermentation? I think all of you from the biotechnology background or has experience run the fermentation, right? From batch, fat batch, and continuous system. Today I focusing on fat batch because we need increase the production of DHA. Then my study is focusing in kinetics modeling of this fat batch fermentation process. Why fermentation kinetic? Because we want to predict how much the production of DHA, and we can profile it by using this mathematical modeling. Okay. When I have glanced through the literature review, most of the fermentation process of this microalgae is normally running out in batch and fat batch fermentation. Okay, and the best yield we found is when we run it in fat batch using the constant flow rate feeding. So my background is a bioprocess engineering that I focusing on the process. Okay, so I come up with the mathematical modeling that's to representing the model, the data, and to validate all this model to the with the data, with the mathematical model. Okay, this is how to I develop the model. This is come from the mass balance. Then after that, we come up with the kinetic model, model fitting, and validation. Okay, this is the model development. When we want to run the fermentation, we have to understand the mass balance of the process. Either it's batch fermentation, fat batch, or continuous. And today we focus in this process. We focus on fat batch. We focus in constant in the this is the mass balance. This is the kinetics reaction that we integrate into the model. Then it's the final model. Then after we get all the
please stop. Your time is over.
Yes.
Your time is over. Please.
Okay. I think from this modeling, we found that all the model we simulate and can be validated with the experimental data. I think if anyone have interested in this kind of modeling, we can contact me. Then I think that's all for my presentation today. Thanks. That's all.
Okay. Any questions, please? So any questions, audience?
What is the design of this fermenter? Is it airlift fermenter or chemostat? Or
this one is a state tank bioreactor, but we running out using fat batch fermentation. That means we need to feed in the substrate inside using the fat batch mode.
Okay, fat batch.
Fat batch mode.
Thank you.
Thank you. I wish to announce that each participant will get a time of about 8 minutes, which includes your question and answer session. So kindly stick to the time, 6 minutes for your presentation, maybe another 2 minutes for the question answer session. Thank you.
Second part is, sorry. So now I request Prakash Katam sir to take up the Take this, sir.
Sir, good afternoon, everyone. Our team is working on 3D printing in pharmaceutical sciences. My present topic is to give a brief idea about the 3D printing and what we are working in our laboratory. Right, left. So many of you know about 3D printing. It's a layer by layer generation of or printing of polymer or any material to make a object. So this is the process of how a 3D printing occurs. We have enormous materials. There is a lot of research is going on nowadays, especially in biomedical as well as pharmaceutical fields. So these are some of the interesting examples for the materials that are used. Typically, there are 7 technologies involved in 3D printing, or it is also called additive manufacturing. As per ASTM standards, we have vat polymerization, binder jetting, metal jetting, powder bed fusion, metal extrusion, direct energy deposition, and sheet lamination. And out of these 3 technologies we are using in our laboratory. In biomedical applications, usually scaffolds, bone scaffolds, grafts, all these are being manufactured nowadays. Even bone replacements using metals, custom made bones are being prepared. This in this, one of the technologies is bioprinter, where it is being activated by laser pulse or thermal, piezoelectric or pneumatic piston, so that cells are printed precisely onto the scaffolds. As far as drug delivery is concerned, the advantage is high production rate and high drug loading, reduction of material wastage, flexibility, and continuous manufacturing. This is most important in drug industry. Nowadays, so far, all the medicines are prepared in batch process, but in the coming days, you will see they will be produced in continuous process. And already there are few drugs available in the market using 3D printed technology, one of which is levetiracetam tablet, dispersible tablet. In the future, you will find that all the drugs will be delivered in a customized manner. So suppose a patient is having identified with a diagnosis, and he has to undergo certain treatment or replacement of organ or tissue or medicine to be given in a precise manner. This 3D printing technology can be used more advantageously. So current research involved, out of 7 technologies what I discussed earlier, inkjet printing, direct wire, zip dose, thermal inkjet printing, all this stuff. This is our 3D printing laboratory. We have a startup also. It is called PrintRx. We are working on 3 technologies here, as mentioned down. So we have one work which we are doing using fused deposition modeling, where we take a polymer like polyvinyl alcohol and ibuprofen, and filament is prepared using a single screw extruder, and the model is designed in the computer, CAD model, and that will be fed to the 3D printer where we'll get these tablets. So these are 3D printed tablets what we have done, and the evaluation is under process. This is another technique. It's our own technique. The actually levetiracetam tablet what I mentioned earlier, the machine cost is around 40 crore. But we are working on a machine which we designed at 10,000 rupees. And soon after patenting, I can disclose in the publication. In this, powder is layered, and on that each layer, binder solution is applied, and again layer of powder is applied until we get the desired thickness of the tablet. So like this, we got 8 mm tablets as well as 30 mm tablets. The main challenges are excipient research is going to be a good advantage for the researchers where we can work more. And actually, we are also working on excipient research using natural carbohydrates. Optimization of 3D manufacturing process is another challenge. And of course, clinical trials is the biggest challenge for any drug delivery system. And also regulatory issues. Finally, I conclude that 3D printing technology will be useful for biomedical engineering or biotechnology, as well as pharmaceuticals, especially for personalized or customized device manufacturing of medicines. These are our publication. This is a book chapter on personalized medication using 3D printing technology. This is very recently, this month we published in this journal. Thank you.
Any questions, please?
Sir, you have mentioned about this natural carbohydrates being used for 3D printing. Now, nowadays, biodegradable plastics are being produced from various organic wastes based on cellulose or starch. So, how are they prospective materials for 3D printing as well?
Definitely, ma'am. Actually, we are working on that also. We have taken cellulose estate butyrate and we coupled with another carbohydrate to make biodegradable plastic. And the same thing we are going to print as a film or a tablet or whatever.
So, is it already in the market?
No, no. We are, it's under process.
Thank you.
Thanks. Usually, in bioprinting, as you see, sir, we use gels, hydrogels. So first we take a beaker, in that hydrogel will be there, and we put the cells in the syringe, and that will be printed precisely wherever you want. So in the 3D structure, for example, you want to print a heart organ, heart. So the same can be applied, and but the advantage with the gels is thixotropic gels. They won't allow the cells to come down. They will suspend like that only. Scaffolds we can use any number of materials like protein, from proteins, agar medium. Yes, sir. Yes, sir. Actually, we are not working into that part basically.
So but you did not try any cell culture?
No. We are not working on the bioprinting. We are working on formulations.
Formulation.
That too, we are using only two technologies so far: fused deposition modeling and the one powder bed inkjet printing.
Stereolithography also you are showing.
SLA also we are using, but we are using for making micro needles. Micro needles.
I'll request you to have a discussion after the session.
Thank you, sir.
Please.
And I thank Professor Kavikishore sir for giving me this opportunity to present this. Thank you so much, sir.
Now I would like to request Venkata Nagamani to start her presentation.
This is JJ Venkata Nagamani, working as ICMR SRF under the guidance of Dr. M. Srinivas Rao, CSR ICT, Hyderabad. Today I am going to present on in silico screening of epitope-based vaccine against Castanor forest disease virus. The main object of the work is to construct multi-epitope vaccine against KFDV and to investigate the influence of immune response towards cell-mediated and humoral mediated responses. And after in silico cloning and codon optimization was examined in order to check the efficiency of the vaccine construct. Introduction to KFD virus. KFD virus belongs to family Viridae. It is an endemic zoonotic disease found in southern parts of the India, and it infects human beings and cause severe hemorrhagic fevers, and the fatality rate is 2 to 10% around the areas of Maharashtra. And KFDV is transmitted by the infected tick of Haemaphysalis spingeria in its nymphal stage. And this is a scheme of study, retrieval of sequence, epitope prediction and evaluation, and the construction of vaccine, and validation of 3D structure of vaccine, and molecular docking stimulation for validation, and codon optimization. This is a T-cell epitope analysis. From the T-cell epitope analysis, we predict the class 1 MHC, nothing but CD8 T-cell epitope prediction and CD4 class 2 MHC epitope prediction. And the epitopes were predicted using immune epitope database and followed by the antigenicity and solubility and allergenicity and toxicity of the peptides are also predicted. And further, the peptides go for screening of proteasomal cleavage and transport efficiency for class 1 analysis, and interferon gamma epitope prediction, and IL-4 releasing interleukin cytokinase inducing peptide prediction, and IL-17 prediction. Further, the epitopes are selected for further analysis. And this is a population coverage of class 1 and class 2 epitope worldwide. From the selected epitopes, 90% of population is covered worldwide. And next we go for B-cell epitope analysis. From the B-cell epitope analysis, by using these servers, we predict B-cell epitopes. This is a molecular docking of HLA class 1 and class 2 MHC. Sorry. This is a molecular docking of class 1 and class 2 MHC selected from previous analysis. And this is a construction of multi-epitope vaccine. From this, beta defensin is added for the induction of immune system, along with the cytotoxic, nothing but class 1 MHC and class 2 MHC. B-cell epitopes are linked with EAK linkers and GPGP linkers and KK linkers, along with at N-terminal 6 histidine tag were also attached. This is a primary and secondary structural analysis of epitope vaccine construct. And this is the 3D structure was predicted by using ITASR and a Galaxy Refined server. This is a 3D structure validation of, This is a 3D structure validation of the multi-epitope vaccine construct through Ramachandran plot and ARAD score. 2D structure. And thus ARAD score is a 3D structure, sir. And this is a vaccine construct that is present in Swiss Institute of Bioinformatics model archive database, and the structure is validated, and this is accessible for users. After constructing the vaccine construct, we need to check whether this vaccine construct trigger the immune response or not by docking with the TLR3 receptor. This is an in silico immune simulation of vaccine construct, whether the immune system is perfectly raised to this vaccine construct or not. This is a in silico cloning of the vaccine construct. First, the codons are optimized back to transcription state, and by adding restriction sites to both sides, and we keep this transcripted nucleotide to a vector, and a perfect vaccine construct was formed with this restriction endonuclease along with the vaccine constructed sequence, and this vector is cloned perfectly, and which is for further analysis. Conclusion of my work is to construct in silico epitope vaccine by constructing with immune inducing peptides of viral proteins, which then combine to form a vaccine construct. And in silico testing is only method we employed to validate the study results, and these results will incite to guide further studies under additional in vitro and in vivo conditions. Thank you.
Why did you prefer TLR3, why not TLR7?
So for viral related immune responses, we choose TLR3, and further more work is there, but we conclude by this part. So
Viral related thing is TLR7.
okay, sir. TLR3, TLR7. Actually, we dock TLR7 too, sir. But because of lack of time, I am not presenting those kind of work, sir. In still under.
What is the size of the multi-epitope?
Sir?
Multi-epitope size, ma?
The size is 704 amino acids, sir.
It is
no.
almost like a protein. Why so big a protein?
Yes, sir.
It is almost like a big protein, ma.
Yes, sir. It is a vaccine construct, sir.
Yeah. So that much of vaccine to prepare it is costly.
Okay, sir.
So epitope, that is polytope or something like that, 3 to 4 epitopes only you have to take. So 700 means what is the size of a hemoglobin?
Size
Okay.
of.
So you take a so big a protein.
Yes, sir.
That much vaccine if you make, it is a costly affair.
Actually, we need for further study we need this. We need to split the vaccines into 3 constructs. Further, in order to validate the construction efficiency, we want to.
Okay. So what about the cloning? In where did you cloning? What type of the plasmid? What is the name of the plasmid?
PGL 4.0, sir.
So it is going to be hosted in a vehicle, no? Cloning vehicle. What is the cloning vehicle?
The cloning vehicle is actually in silico, sir. Just mammalian expression system, sir. It is clone.
How do you perform immune response in silico?
By using C immune sim server, sir. Yes, sir. Yes, sir.
So what was the injection, means how much the injection type, means what you put there on in sim, means injection portion, let's say 2 times, 3 times, how much?
Actually, it is 10 times, sir.
10 times. So
Yes, sir.
a person have to take 10 times a vaccine.
Sir, actually, the work is not completed yet. We go on further analysis.
Okay. So if you saying the 10 times, then have you saw in that means simulation result that after 10 times means when you putting 1 times, then whatever the curve is there, so curve will be low, right? And
Yes,
then
sir.
after when you're putting the second time, then curve will be the high. Okay. So like that, if you put the 10 times, have you checked that one?
No, sir. Okay.
Okay. Thank you.
Now I request Dr. Nagraj to start his presentation.